It was established as Pathshala for the Birlas in 1901 and was given the status of deemed to be university in 1964. The Department of Pharmacy of BITS Pilani, established in 1950, is one of the early schools of pharmacy in India. It has been ranked 6th by NIRF in pharmacy category. The department offers on-campus B.Pharm, M.Pharm with specialization in pharmaceutics, pharmaceutical chemistry and pharmacology as well as PhD programs. Department also offers M.Tech and PhD programs for the working professionals of renowned pharmaceutical industries. We provide to students an exhaustive exposure to various practical aspects of research methodologies, instrumental techniques, courses in emerging areas, and industrial operations. Department has a vision to become a global center of excellence in pharmaceutical sciences and research. To achieve this vision, we have faculty members who have research bent of mind and expert in their own areas. Department has 3 thrust areas, namely pharmaceutics, pharmacology, and pharmaceutical chemistry.
So in pharmaceutics and pharmacokinetics, faculty members are actively focusing on the key areas involving the development of controlled release dosage forms as well as novel drug delivery systems, including polymeric nanoparticles, solid lipid nanoparticles, polymer drug conjugates, and liposomes, to name a few. Further, our faculty members have expertise in pharmacokinetics and biodistribution studies, PKPD modeling, and IVIVC. We have a dedicated industrial research laboratory that houses requisite facilities to further our research programs. These programs are sufficiently funded by the government as well as pharmaceutical industries. Herein, equipment such as extruder spheronizer, multi-station mini press, rapid mixer granulator, spray dryer, etc. are available for solid dosage forms development. The facility for the advanced drug delivery systems includes high pressure homogenizer, high shear homogenizer, sonicators, etc. For the characterization of the developed formulations, we have the analytical equipments housed under one roof. To further enhance our analytical capabilities, a highly advanced UPLC triple quadrupole MSMS facility is established in the Industrial Research Laboratory. Further, a dedicated mammalian cell culture facility consisting of Class 2 Type A to LAFs, CO2 incubators, inverted fluorescent microscope has been established for the evaluation of the developed novel drug delivery systems. As a whole, our research is aimed to improve the efficacy and safety of the drug molecules by utilizing the new delivery strategies.
Faculty in the area of pharmacology are currently working on understanding the molecular mechanisms in the development of metabolic diseases and neurological disorders. To conduct a cutting-edge research, we have CPCA-approved Central Animal Facility. The facility houses animals like rat, mice, guinea pigs, and rabbit. The facility is approved for breeding as well as housing the animal for research purpose. In addition, we have pharmacology research lab and cell culture facility for carrying out advanced research in the areas of preclinical pharmacokinetics, bioavailability studies, pharmacological screening of various synthetic and natural origin drugs. The pharmacology lab had sophisticated instruments such as autoanalyzer, 2-chambered automated organ bath, laser Doppler, non-invasive blood pressure recorder, RT-PCR, electroconvulsometer, and other equipments which are used for the other experiments. The lab is upgraded with video documentation system for various animal behavioral studies. The facility is geared to take up various industry and government-sponsored projects in various preclinical areas.
In the pharmaceutical chemistry specialization, the main focus of research includes design, synthesis, and in vitro in vivo evaluation of new chemical entities against neurodegenerative disorders such as Alzheimer's, Parkinson's, rheumatoid arthritis, infectious diseases such as HIV and its allied opportunistic infections like tuberculosis, malaria, leishmaniasis, and other microbial fungal infections as well as cancer. We also have natural product research area. So in that, main focus of research includes synergy guided extraction, isolation, purification and evaluation against various metabolic disorders such as obesity, diabetes, etc. In order to perform the designing part, we also have a computer-aided drug design laboratory with the various advanced softwares such as molecular modeling software such as Schrodinger Glide, Biosolve IT, CSAR, StarDrop, VLife MDS, Autodoc Vina, Gromax, etc. In order to carry out the synthetic synthesis, extraction and isolation related work, we have well-equipped, sophisticated, dedicated laboratories with facilities such as parallel synthesizer, flexible microwave synthesizer, rotary evaporator, etc. For the purpose of characterization, we also have HPLC, HPDLC, flash chromatography, NMR, LCMSMS, GCMSMS, etc. For in vitro evaluation, we have multimode spectrophotometer, and for in vivo evaluation, we will take the help of pharmacology faculties.
Central Analytical Lab at BITS Pilani houses several sophisticated instruments and caters to the need of various departments like pharmacy, biological sciences, chemistry, and physics. The list of equipment include high performance liquid chromatography, electrophoresis, UV visible spectrophotometers, flash chromatography, and elemental analyzers.
Apart from the research facilities and labs in the department, the Institute has also been supporting us generously in our research endeavors by establishment of a sophisticated instrument facility. The objective of the facility is advancement of research in the Institute as well as in other nearby universities at very nominal charges. In the facility, we have several state-of-the-art high-end equipments including surface enhanced Raman spectrometer with atomic force microscopy, gas chromatography, mass spectrometry, 256 core high computing computer cluster facility, field emission scanning electron microscope, NMR, confocal microscope, multi-parameter nanoparticle analyzer and impedance analyzer to name a few.
Department is actively involved in collaborating with national and international research organizations with pharmaceutical industries. Department is actively involved in industry-academia linkage to generate industrial relevant knowledge at affordable cost. Department has collaboration with various pharmaceutical industries such as Dr. Reddy, Sun Pharma, Intas, Ipca Lab, Slebeck Pharma, Colorcon, Imami, Lupin, and many more. The department has successfully transferred technologies to industries such as aceclofenac, control list tablet, which named as a brand name as Girodol CR and Movan SR to Ipca Lab. Also worked on liposomal drug delivery system for dopamine for the brain delivery and delivered and transferred to Torrent Research Center. Recently, department has also transferred one technology that is called InstaDerm for fast healing wound care product to the Elicon Medical Private Limited. Recently, Department of Pharmacy is involved in various startups that is facilitating to the students and faculty startup. One startup that is Innovate Healthcare and another is Nanobrid Innovation Private Limited that is founded by department's faculty members for the platform technology for the drug delivery with industrial feasible process.
Department of Pharmacy, BITS Pilani provides overall development and empowers students in all aspects. We have world-class curriculum and faculty. Our pharmacy programs are designed to give our students a balanced education with theory as well as practical knowledge. Our practice school program ensures that every student has spent over 7 months
So, hello, Professor Mahato.
Yeah. So, hi, Deepak. I will remove mask in a few seconds. Let me first. So, should I share the slide or no? How
Yes, you can.
Yeah,
you can share the slides, Doctor.
Yeah,
I will be starting in next 10 minutes.
At the moment, Bidendra is around here. So let me. So how to go back? No, how to go back to. Just a second. This one. Present now. Present now. Entire screen, right? This one. And then you said screen to where is screen 2, right?
Yeah, sure.
And share. And then I have to, of course,
Make it full screen.
can you see it now?
Yes, Dr. Mathur, we can see.
Yeah, so one more thing that I need to do before you go. I need to make, yeah, okay, so yeah, I'm ready now, so tell me, yeah, you can be maybe a couple of minutes, yeah, if not, then you can go. Yeah, so Deepak, let me know what I need to do and when I need to get started. I know we still have a few minutes, right? So do you want to check the slide or should we find how you want me to work?
Yeah, this is fine, Dr. Mathur. I mean, we'll start in another 10 minutes.
Yeah.
Just to check that whether your slides are coming fine. So I thought to
Yeah,
have
so let me run the slide quickly.
Yeah.
Can you see the slide?
Yes, everything is coming fine, Dr. Mathur.
So I don't have to go through all slide now, right?
Yeah.
Okay. So you have more people. I saw a lot of facility. Yeah, you guys have a lot of facility. So how many students do you have in your university, in your college? Hello?
Yes. Yes, Gaikwad sir.
Sir is asking some questions to you. How many students are in college?
Yeah, I wonder if you could, to utilize the time, maybe you could share a little bit about, because I didn't get a chance to watch everything, but I did watch a little bit. About your university and in particular about your college, you call faculty, right? Faculty of Pharmacy.
Yeah, Hemant sir, are you there, sir? Hemant sir?
I think he is
Yeah,
ठीक है दीपक still you can
Maybe सर आप brief कर दीजिए. अनिल सर आप कर दीजिए brief.
Okay.
Yeah.
Good morning, Professor Mathur.
Good morning.
I'm Gaikwad, and I'm currently the pharmacy faculty here. So here, the pharmacy is a department, BITS Pilani is an institute. So overall, there are no separate segregation like whether it's pharmacology or like this. So overall, Professor Hemant joined. He's the current head of the department. Maybe he can speak about the department.
Oh, I see.
Yeah. Hemant sir.
Yes, Anil.
Yeah, Professor Mahato is asking a little bit about department. So we were waiting for you, so I started it in between. So you please continue with something. He's asking about programs, how many students, etc.
Yeah, if time allows, it is all up to you, no? I think the time is on. We have 4. I hope it is not a stock, right? That's material. 3 minutes.
Yes. Hemant sir, I'm audible, sir?
Yes, Deepak.
Yes, sir. So Professor Mahato was just asking for the department brief out, just since we have a few minutes to go. So
Yeah, if you have time. Otherwise, it's fine. I mean, we can chat later. That's also fine with me.
Okay. Actually, we are also running live on YouTube, and I just checked people are joining there also. So we have quite old history. We started in 1950. Ours was probably second or third pharmacy institute in India, and and over the years we have expanded our curriculum also, programs also. Like initially it was I Farm, then B Farm, then we started M Farm. Then 2008 we started M Farm, specialization in pharmaceutical chemistry and pharmaceutics. Recently, like this year onward, we have started M Farm pharmacology also. We have 16 faculty members here in Pilani, as well as around 12 faculty members in Hyderabad campus. So we have pharmacy department in 2 campuses, Pilani as well as Hyderabad. And in India, we are ranked 6th by the NIRF, that is Ministry of Human Resources, that ranking was there. So we are 6th in India. We missed out on, you can say, 3rd rank by just 1% point.
Oh, I see. That's wonderful. Okay.
Just by 1% we are missing, otherwise we would have been 3rd in India. So our faculty are actively engaged. This year alone we have published more than 52 research papers, got 16 projects, 5 group patents, and as you are aware, Deepak has started one startup spin-off, you can say. Another is another faculty has started another spin-off. So quite active department we have.
Oh, wonderful! Yeah,
around 44 research scholars, that is PhD students. M-Farm strength is new admissions 53, and in the previous batch of I think 45 we had. So,
so overall, you have very good and very active department, I guess.
Yes. And BITS gives a lot of flexibility to students. They can do off-campus thesis, they can do vertical transfer, horizontal transfer. A lot of opportunities are there for the students. They choose electives in each semester. Right from the beginning, we give them options to choose the electives. They can either go to practice school, that is doing the job, doing the work in industry, or they can continue dissertation on campus. Flexibility is there in BITS.
Okay, that's wonderful. Okay.
So I think should we start? It's 9:30.
Yeah. Thank you for the update. Yeah, thank you. Yeah, we can get started.
Yeah, we can talk later, Dr. Mahato.
Sure. So should I begin talking, or how should I go?
yeah, I'll introduce, sir.
Yeah. Okay.
So thank you all for joining in. So today we have the second day of our conference, and yesterday also we have seen speakers from varied areas, both from international and India, from academic as well as industry. And today also we have 6 speakers with us. And the first one, we have Professor Mahato from University of Nebraska Medical Center. So it gives me immense pleasure to introduce, as I have been trained in his lab. So Professor Mahato is a professor and chairman at the Department of Pharmaceutical Sciences, University of Nebraska Medical Center, Omaha. He was a professor at the University of Tennessee Health Science Center, research assistant professor at the University of Utah with Professor Sungwen Kim, senior scientist at Gene Medicine INC, and as a postdoctoral fellow at the University of Southern California in Los Angeles with Professor Vincent Lee, Washington. He has also worked in Kyoto University with Professor Mitsuru Hashida. He received his PhD in drug delivery from the University of Strathclyde, Great Britain, and BS from China Pharmaceutical University, Nanjing. He has published 160 peer-reviewed papers, 24 book chapters, hold 3 US patents, and has written 9 books and 11 journal issues, with a total citations of more than 11,000 and has a H index of 62. He is an associate editor of the Journal of Neuroimmune Pharmacology, was a feature editor of pharmaceutical research from 2006 to 2013, and is an editorial board member of 8 journals. He is a CRS Fellow 2011, APS Fellow 2010, is a permanent member of BTS's NIH Study Section, and ASGCT Scientific Advisor, Known Viral Vectors from 2006 to 2009. He is applying sound principles in pharmaceutical sciences in the context of latest advances in life and material sciences to solve the challenging drug delivery problems in therapeutics. His areas of research include drug delivery and targeting of small molecules, microRNA, and genes using novel polymeric and lipidic carriers for treating cancer with a focus on pancreatic cancer, prostate cancer and melanoma, liver fibrosis, and diabetes. And with this introduction, I invite Professor Mahato to deliver his talk and give his insight in the field. Now over to you, Professor Mahato.
Yeah, first of all, thank you, Deepak, for your nice introduction, and also my thanks to your department chair, Dr. Jadhav, and all students and faculty for finding time to attend and listen to my presentation. And especially for me, it really gives me a great pleasure because for some of you may not know, but I was really born in Nepal. You might have heard Janakpur, which is it's Mithila, right? And then from there I went to China. So naturally I can speak Hindi, I can speak Chinese, I can speak Japanese, you name it. But with the interest of time, I really want to share with you what I learned in this journey. And so one topic I really want to share with you about how can we really combine small molecules and nucleic acid drugs for example, microRNA, which is non-coding RNA for treatment of different diseases. But for this presentation, I have focused mainly on pancreatic cancer. So first, let me tell you why I'm so interested and why especially young people there should be interested, because the work we do, basically we really apply nanotechnology. As you know, nanotechnology you can use in anything. It's a buzzword now. You can use, but for medicine, especially in pharmaceutical science, we can really use it wisely for prevention of the disease, for the treatment of diseases. You can use for diagnosis. And really the overall goal of our purpose really is very simple. Because, we study pharmaceutical science. Some you may say that really I really study to buy a good car. Some may be dreaming to have a big mansion. But really ultimate goal is really how can we pharmaceutical scientists improve the quality of life and lifespan of a patient. And this is our ultimate goal, and that's what we are trying to do. And we believe we can utilize nanomedicine approaches to achieve these goals. So my lab is really very diverse, simply because I have very diverse expertise. Certainly, I always believe in designing career, like in terms of polymers in the past, even now we talk about lipids. Sometimes we design peptide. When I say peptide, this career design means we try to synthesize in the lab and for different purposes. But at the same time, I always believe that really disease can be treated better if we can treat at a molecular level. So therefore, you could really also use gene expression systems to express or silence certain genes. And for this, I've done extensive work to really design different expression cassettes to express genes like a hairpin RNA or even microRNA. You can do that. And ultimately, by training, we are pharmaceutical scientists. So naturally, we do the formulation and we assess in vitro as well in animal model in terms of cell culture work, pharmacokinetic, pharmacodynamics, on and off, we also use pharmacogenomics because there is a patient-to-patient variation of the same therapeutics have different response, and why it is. So that's we also try to do it, and we do this really in three different disease model. One is liver diseases, and one is in diabetes, and also in cancer. So today I will talk more about cancer. And in each case, one of the key issue probably many students there will be interested because our goal is really to deliver drugs. And drug, when I say the drug can be anything, a small molecule, macromolecules. And in each case, really you can deliver in two ways by using particulate systems, or you can use by conjugation approach, so-called micromolecular systems. So in particulate system, my lab is really involved with both, like liposomes, our nanoparticle, of course, our micelle, and each has own advantage, disadvantage. And with the interest of time, I'm really leaving up to you to learn about this. But occasionally, we try to really conjugate our drugs or nucleic acids to certain polymers or lipids using pH sensitive or some kind of cleavable spacer. And this is really for two reasons. One, to really simply increase their stability, but also to increase the target specificity. And this all really we have richly expressed in discussed in different publication from my lab, and these are some of the examples of the books or the theme issues. Some I've listed, some not. And I would really encourage, especially students and young faculty, to really to find some time to look into this. You may find something of interest, and I'll be happy to even send you a copy if necessary. But today, really, I'm trying to share with you about the tumor. And the tumor, when we talk about the tumor, as you know, there are different types of tumors. And I'm trying to tell you about pancreatic tumor. But here, the tumor, what you will have is you will have different cell types. And among them, of course, there will be tumor cells. There will be also macrophages. There will be extracellular matrix. And of course, tumor, you will have also hypoxic conditions. And there will be high interstitial pressure, so which will retard the distribution of drug in the tumor. And quite often, especially in case of pancreatic tumor, there will be also dysplasia because why this is so important, because desmoplastic tumor does not really allow efficient distribution of drug to the pancreatic tumor. So therefore, this is a big issue and need to be addressed. And there we believe our nanotechnology approach can be used to really address this. And of course, we have to really address a lot of biological cancer biology issue. And if you're interested, you're welcome to really visit our special issue to address some of these concepts. And first, up to really 2006 or so, I was really more, 2007, you can say, I was more interested in nucleic acids. Basically, I was working on genes like virus and non-viral gene therapy. I was working a small, siRNA. But I really, one student visited from Kentucky. This guy is here. His name was Michael Dangwa. And he really wanted me to really work on work on a small molecule. Michael Dangwa now is a department chair in Chicago. And he really wanted to begin the work with the polymeric micelles. And this is the benefit he shared with me to really win me back, because I was really very reluctant to work on this. And basically, you can use the micelle first. It's very simple, as you know, the surfactants like properties. It can is small in size. It can have high drug loading. You can really control the release of the drug, our site specificity, and so on. So therefore, I got interested, and also most importantly, from beginning, I was really less interested in using commercial polymer, and therefore Michael really came up with the idea to modify the polymer by introducing carbonate unit into this. And this really helped two things. First is increase the drug loading, and also the degradation of carbonate unit really is not acidic. And therefore it became very attractive, and we did some work on that. And students, if you're interested, really visit that. But of course, in between many things, and of course, we did a lot of work initially with a small interfering RNA. And I asked my former student, Dr. Puncham, who is now full professor at University of Missouri, Kansas City. He's very rich, a lot of grants. So Kuhn really tried to focus on siRNA. But at the same time, I had a postdoc who joined in 2008 from China. And he was very much interested really to initiate microRNA, which is non-coding RNA. And he is now a really full professor at Tongji University in Shanghai. And that's why we try to compare why should we really switch from one area to another, why from siRNA to microRNA. And he impressed me because the number of publications, as you could see in this slide, is significantly higher and very rapid increase for microRNA, whereas the number of publications for siRNA beginning to slow down. So I thought it's a good area to get started. And then, of course, with some time, there was another postdoc who is, I think, now in India, Dr. Saurav Singh. I think he was working in Novartis, India. I don't know where he is now. And at the same time, I visited at that time, I think it's in India, and I met Deepak and Anupa, and at least I win back Deepak to come to my lab as a visiting student. And the reason was very simple, because I like the guy, and I thought he should really spend some time with me. So Deepak and Anupa joined, and they really began to come up with the idea. I'm trying to make it short, really to combine a small molecule with microRNA. And basically, they were interested in gemcitabine. And as you know, in this slide, I very richly explained the reason. And with the interest of time, I just want to tell you a couple of things which draw my attention. One, because whether it's gemcitabine or any other drug, if you give high dose, it will really lead to systemic toxicity and also really cause chemoresistance. And therefore, we thought we can combine microRNA, which can really inhibit chemoresistance, and in return can chemosensitize the cancer and cancer cells. And then later also Birendra Kumar, he got involved. He was a student at that time. Now he's an instructor. He should be promoted to faculty here. And they begin to work on that. So, but only my request to Deepak at that time was really, I want to deliver both microRNA and the gemcitabine together as a single formulation. So that's why Deepak really introduced this cationic chain into this polymer, which will allow the ionic interaction with the microRNA, and also he conjugated gemcitabine to the polymer. So basically, it formed the polymer drug conjugate, and because of ionic interaction, microRNA got protected because it got encapsulated in the core of the micelle. Because once you interact, ionic interaction, they basically insolubilize microRNA. So with this concept in mind, they did a lot of work, and this is the kind of design element. And of course, they have a very nice publication biomaterial, and you are more than welcome to read on that. And but I also was really interested to see really does it really help bioconjugation in terms of biodistribution, and if so, really it can also does it help in terms of really inhibit the metabolism or slow down the metabolism of of gemcitabine. Here, gemcitabine is abbreviated GM, and there was the Krishna Patel. He was from Nepal, but he has done PhD from South Korea and postdoc in Japan. And he joined my lab, and he really did a biodistribution in orthotopic model. And he clearly saw that first, of course, it can really increase the drug distribution, and also at the same time really decrease the metabolic conversion of gemcitabine. And the next slide, as you could see it affect in the tumor in the ear. The distribution to the tumor was increased by 2.5-fold after systemic administration. And keep in mind, these are not sulky tumors, they are orthotopic tumor. So we felt, and of course measured by, so it was a good move. Then after that, of course, we did a lot of work. We measured the tumor, but we realized there's some decrease in tumor size, but really still tumor decrease, inhibition of tumor growth is not as good as we like. And therefore, I introduced another fellow. He is Gautam Mandal. He is now in another university. And Gautam, he was a postdoctoral fellow. And what he did is he introduced targeting. So he basically attached targeting ligand. Here he used antibodies to target EGFR receptor, which is overexpressed in pancreatic tumor or any tumor per se. And he saw, he demonstrated that, of course, it first increased the distribution, and this conjugation can really inhibit the, what you call, inhibit the tumor growth. And apart from antibodies, he also used GE11 peptide, which also targets EGFR receptor, and he saw fairly good benefit in terms of inhibiting tumor growth. Then now question I wanted to tell you all for educational sake, especially for our students, why we are really interested in microRNA. Why not bringing a small, another drug? And this was really discussed. First, we discussed in one editorial by my former student, Michael Dangwa, which, as I told you now, is a department chair. So he, at that time, really explained the microRNA has very unique properties. Because first, microRNA, as you know, it really affect EMT, epithelial to mesenchymal transition. And that EMT transition, as you know, quite often the cancer stem cells really our cancer stem cell phenotype quite often being affected or converted also due to the aberrant expression of microRNA. And other thing which we also realized that microRNA really deeply involved in chemoresistance. And therefore we felt that if we could target not only the bulk tumor, but also cancer stem cells, CSC, cancer stem cell, and EMT cells, then will be good because they are chemo-resistant cells and have metastatic potential and express a high level of hedgehog ligand, which I will explain you later. Also we clearly realize that this regulation of microRNA is really associated with tumor initiation, differentiation, EMT phenotypes, like cancer stem cell proliferation, etc. And therefore, use of microRNA becomes very, very important. Now, question is which microRNA will use? So, a lot of people in the pharmaceutical field, basically, you know, by nature, we are people try to mix up, right? Mixing to prepare compounding. But from beginning, I was really reluctant to do that. So, we did a lot of profiling. So, we did profile the microRNA expression in different cell types, also patient tissue. And we realized that some key microRNA are upregulated and downregulated, and then we use bioinformatics to really decide their target genes. So here, one microRNA we got interested was microRNA 205, And the reason we were interested, because as you could see it, when we did bioinformatics with software, it's a free, done mainly by students. Really, I don't even know how they do it, but they really came up that this microRNA 205 affects a lot of things. It really affects drug cancer cell invasion, migration, metastasis, then the radio resistance, chemo resistance, you name it. So therefore, it became a very important target microRNA. And this microRNA, as I told you, it is downregulated in the pancreatic cancer, right? So therefore, we use microRNA mimic. And this first was really done by Saurabh, who even before Deepak began working on this. And he clearly told us that when we use resistant, gemcitabine resistance cells and combine and then treat with microRNA 205, then it will chemosensitize the cells, and that will lead the killing of cells with increase of gemcitabine concentration. So clearly so that, and also the sort of really clearly showed that microRNA 205 has significant effect on certain genes which are directly associated with chemoresistant, like OCT34 cells, OCT34, like CD44, like TOB, especially TOB. TOB is directly related to chemoresistance. So there was a very good rationale behind of using microRNA 205 in combination with gemcitabine. And this, of course, this is the formulation. I think Nukma and Deepak did it, and they confirmed that microRNA need to be dissociated. And for, as you know, microRNA, you cannot see it, so they did the gel electrophoresis to confirm that it can dissociate, especially in presence of DTT, and suggesting that really you can formulate both at the same time. And then, of course, they showed even in animal model that their combination indeed will have some benefit. So before but one problem with here was, as you know, that a couple of issues I wanted to share. First, gemcitabine is a water-soluble drug, is rapidly metabolized, and since it's a water-soluble drug, we have to really conjugate to the polymer. So then Birendra got involved, and he suggested, Dr. Mathur, rather than using gemcitabine, we should use Hedgehog inhibitors, which he was working with liver fibrosis, because Hedgehog inhibitor is insoluble in water. So basically, you could really encapsulate. So you don't need to really conjugate to the polymer. And also he suggested rather than using 205, we should use lead-7B, another microRNA, because that is probably more potent than 205. And that's what he thought. And so again, we did, and the reason we selected Hedgehog inhibitors simply because, whenever you have inflammation, whether it's cancer, whether liver injury, there will be upregulation of a hedgehog ligand, like a sonic hedgehog, like Glee, like Smoothen, and this we really saw Western blot as well as real-time PCR, and therefore inhibition of this really lead to a benefit. And some of you may remember that in fact there is a science paper that the clinical trial with Hedgehog inhibitor failed. And the reason they failed because they use a free drug, and free drug, if you use a very high dose, then naturally it will cause toxicity and so on. So that's why we thought we should formulate this and they had also used a single drug, but in our case we used with microRNA lead-7B, and then we did show that indeed this is a beneficial to do that. And of course, we measure a lot of other genes, and we check with the different pancreatic cell line to really convince that really lead-7B is down-regulated, and therefore its combination with GDC, which is a Hedgehog inhibitor, will be beneficial. So we did, of course, a lot of work there, and we could easily formulate this together. And the presence of microRNA had little effect on GDC release. And of course, we check in animal model, and as usual, we measure tumor size and so on, and saw that tumor growth is slowed down. But as you remember, gemcitabine is a commercial product, number one. And the gemcitabine, another issue is still cause resistance, means after repeated use, it doesn't work well. And the binding efficiency to mutated smoothing is very, very poor. So because of this reason, gemcitabine is not as good as being promoted. So we thought maybe we should do something about this, right? So then I had, being a department chair, you have one benefit because you know the people who really need some work. So I met Yu Xiangdong, who is a research faculty, and also is from Nanjing, so where I spent some time. So I asked Yu Xiang that, Yu Xiang, I'm working with hedgehog inhibitor. Can you help me design this with some modification? And I'm not really pure chemist, but I draw something and I said, this is this, and can you really make some changes? Then Yu Xiangdong really designed a lot of analog of GDC. Some I'm not showing you simply because Deepak told me that you want to put in YouTube or something. So unpublished thing I didn't want to share. But MDV5 I'm sharing with you because this is already published, and in fact is now being manufactured by one of the company in US. Of course, I don't have patent right because I published without that, but the company, in honor of our contribution, they still kept our name MDV5 in their product. So that's good. So we did it, we modified this, and compared to gemcite, to GDC, naturally it is much more effective in terms of inhibiting hedgehog ligand. And most of the Western blot is done by really Bharti, which is from Haryana, I guess, or somewhere from Punjab, and he's a graduate student here. And he worked closely with Virender, and you could see that whether it is on the silencing or the IC50 value is much better than GDC. But I want to really draw your attention. It's not
Good
so
morning,
common.
everyone. Am I audible or some echo is coming?
Oh, echo is coming? I don't know. I am not keeping anything on. Everything
This is fine, Professor Madhu. This is fine.
not from our side. Maybe one of you might have kept your speaker on, but really not from here.
That's okay. That's okay,
Yeah.
Professor.
Yeah. So let's move on. So what we did, we designed a lot of them, and I show you a couple of data. And one data, just to, of course, we did a lot of things. Most of the things are published. So interested student or faculty can really kind of also really check our literature. But one thing I wanted to tell you that MDV5, which is a hedgehog inhibitor analog of GDC, naturally is much more effective than GDC alone. So that's the main message. And we published a scientific report for cancer. Then we published diagnostic for liver fibrosis and so on. And I don't have all in my head, but you could really dig out our presentation. But I want to really share with you the today topic, which is something different with the time I have. And I told you why I'm trying to show this to you. The first thing is Sorry,
I missed that. Could you say it again?
Yeah, I'm sorry. So first is the redox sensitive.
I'm not sure I'm here.
Oh my God. Yeah, I apologize. So redox sensitive polymeric system, we thought we should use it. And also we thought we should utilize hypoxia. Because as you know, pancreatic cancer quite often undergo hypoxia, under hypoxia, right? So the hypoxic condition is one of the key issue, and another is of course dysplasia. So this work really initiated by Vinod. He is now at Pittsburgh, And then just like Deepak, when I went to India, I stole Deepak back to US, brought him US. Same way when I went to China, which I go almost every year, I met Xiaofei, this girl, and her husband, at that time boyfriend. So they were graduate students. So I asked them, okay, they should come to US as a visiting student. They joined my lab as a visiting student, and now, of course, they are visiting a scholar. They finished PhD. And so she really worked basically on this project. And then this is the Feng Lin, my former graduate student. He's very good polymer chemist, and he helped us design the reduction city polymer to deliver this. And I'll tell you as we go around. So question is first, why should we use 519C microRNA? Why not microRNA 205 or lead-7V? The reason is very simple, because microRNA 519 has some of the properties very interesting. So first, of course, just like microRNA 205, microRNA 519 is down-regulated in tumor. And also there, and the microRNA 519 expression is severely decreased, is reduced in cancers, pancreatic cancer cell line, and like here, Miyapaka, resident Miyapaka 2 cell line is certainly very, very low. Capans 1 has also very low expression, and another thing we found, as you could see, this was confirmed with stone blot as well as real-time PCR. So we felt that we can use this, their combination. And I'll tell you why we think so in the next slide. So here, as you could see, first, when we created under hypoxia condition, right, and we use a scrambled microRNA, and we use microRNA 519, we clearly saw that microRNA 519 has beneficial effect in terms of inhibiting the migration of the cells under hypoxic condition as well as under desmoplastic condition. For desmoplasia, to create desmoplasia, we mix our cells with hepatic stellate cells, which is the cells present in pancreatic, the human pancreatic stellate cells. So pancreatic stellate cells present in pancreas, and these are the cells really produce ECM, excess amount of ECM, including collagen, and that cause desmoplasia. So that's why we mix the cells with this pancreatic stellate cells, and under hypoxic condition, as you could see it, a scrambled microRNA has no benefit, but when you use microRNA 519C, this is a benefit. This is a really very extensive work. I'm just sharing with you a few slides. This will be out next week. I think November 11 in Science Advances. So students, if you want to read it, you could do it, because I'm really not allowed to put the citation before November 11. Is advised by the journal, but you will have access from November 11. So the other thing is the other reason we are interested in microRNA 519, because microRNA 519 inhibits glucose metabolism to really inhibit gemcitabine resistance to pancreatic cancer cells under hypoxic condition. And why I bring metabolism? The metabolism is also very, very important, as you know, because of this. In fact, I've asked Dr. Mittal, Anupam Mittal, to really work with me for one special issue we are working, so-called diabetes-associated fibrosis. So she's trying to create one special article on that, because diabetes, in cancer is very, very important, and so is in case of fibrosis. And when you talk about diabetes, then naturally metabolism, glucose metabolism is very, very important. And that's what we wanted to do. So as you could see it, under hypoxia condition, especially under hypoxia condition, microRNA 215 really inhibited GLUT1 expression, whereas the scramble microRNA has little effect. And another good thing is, and I apologize for this slide, we mixed some of the writing, but basically what you can see is the DCTP level was also significantly decreased by the treatment of microRNA at 519C, but with the scramble microRNA had little effect. This is under hypoxic condition. And the other benefit, as you could see, when we and we also saw that really it affected on many other important genes related to glucose metabolism and so on, and also it has effect on deoxygenosis, what you call this uptake, deoxyglucose uptake. As you could see it under hypoxic condition, this microRNA 519C significantly decreases its uptake. And this has effect because this is competing the uptake by gemcitabine. So it is, and that's why this become very, very important to really to use microRNA 519C to really enhance chemoresistance and also inhibit so-called inhibited this glucose metabolism. So with the interest of time, the other thing you could see it, and we clearly saw that microRNA 519C and gemcitabine have synergistic effect in terms of inhibiting pancreatic cancer cell proliferation as well, and also in promoting apoptosis. So I have really excluded many slides related to apoptosis, but I do want to share with you one, as you could see on the left side, as we combine microRNA with gemcitabine at different concentration, as you could see, the combination has significant effect in inhibiting microRNA, inhibiting cell viability. I apologize for this, this statistics, all symbols mix up. And also we created 3D model, and 3D model, as you could see it here, so like tumor explored we created, and tumor explored is the control non-treated tumor explored. This is the tumor explored treated with microRNA 519C alone, very few cell death, apoptotic cells with red cells, red dot represent the cells undergoing apoptosis or dead cells, and this is with gemcitabine. But with the combination, as you could see, combination of with the gemcitabine and microRNA 519C, as you could see, in tumor asteroids, majority of cells are really undergoing apoptosis. So you could see in red color, and that clearly, and of course we also did like cell cycle, a lot of apoptotic genes we measure, and we saw similar effect. The other thing I wanted to share a little bit, changing the gear, like microRNA, right? So first the challenge is selection of the right microRNA. And as I said, depending on the investigator, some don't want to waste time, so they just buy from the company, and some really do the profiling, right? But the other key thing is, just 1 second, because I want to see the time. Yeah. So the other key thing is, so when you use the if suppose even if you select microRNA, the right microRNA, is it done deal? Answer is no, because microRNA is a nucleic acid, right? And especially single-stranded non-coding RNA, fairly unstable. very, very unstable, especially in vivo's situation. And therefore, the challenge is to how to do the backbone modification, so-called chemical modification of this microRNA, which should really not have any adverse effect in terms of its hybridization or so-called its functionality, right? But it should increase the stability. So we really did a lot of backbone modification. And I'm showing you one with the interest of time. So one modification we did it, basically, we introduced two methoxy, and also partially phosphorothioylation. So these two modifications in the same backbone had a lot of benefit. First, it could significantly increase the stability. As you could see it when we incubated in the serum, microRNA without modification degraded very quickly within 6 hours, almost all degraded. But with modified microRNA, even in 24 hours, most of them remain intact. And now, of course, some students may ask, why not simply using so-called PS or fully phosphorylated microRNA? The reason is when you use fully phosphorylated microRNA, which I did long time back as oligonucleotides when I was still in Japan, that they really increase significant binding to the protein and plasma protein, and also it affects hybridization, and that's why it's not good to have fully phosphorylation. So that's why we did this kind of modification. And this modification, another benefit was this really also in fact to some to have slightly benefit in terms of increasing the efficacy of microRNA. And most importantly, it really decreased the immunogenicity associated with microRNA. Because microRNA, you don't want to use a microRNA should really cause any immune reaction, and which you could see here. Let me have just one or two slides. Then, of course, we did the formulation and characterize in terms of particle size, like surface morphology, atomic force microscopy, drug release, etc., etc. We all did it. And then, we check in animal model. We saw that indeed a redox sensitivity is important, and it has really decreased the tumor size significantly. And this is important because when you see in this slide that our polymer indeed it is pH sensitive also, so it can control the release or maximize the release of microRNA after internalization to the tumor. So finally, I want to escape this side. Finally, I want to end this talk. All this work, what I really want to tell the students and some faculty alike, collaborative effort is very, very important here. So when I meant collaboration, sometimes we do collaboration for the sake of collaboration. And that I'm totally against, because don't do collaboration simply because he's your department chair or he's your dean. Again, I'm not against Dr. Jadhav, don't get me wrong, but do collaboration only if it is a genuine reason. For example, like Pixar, when you combine like Toy Story, right? Music and engineering combined together, right? Then you have big shift in terms of improvement, and this is what is needed, because what young minds should think of is 10x or beyond really improvement when you do this kind of method rather than really one-fold increase or 0.5-fold increase. That is not really going to do anything. So therefore, I do encourage you wise collaboration. And finally, what I would suggest, the era of single using monotherapy is really gone. So quite often you have to do the combination therapy. And I do encourage you to consider microRNA. And when you do that, really don't simply buy microRNA. Try to find out which microRNA you need to do. You may have to do microRNA profiling. You may have to learn a little bit RNA sequencing so that really pinpoint the exact microRNA. And I would prefer to begin with some patient sample, and then you do testing, and then if necessary, again you reverse back to make necessary changes in your formulation and so on. So this all work really done by, not by me, mostly done by a lot of people. Some of the names are missing, seems they didn't get time to really acknowledge all. But I also like to thank the funding agency, of course, university, and NIH, Department of Defense, Buffett Cancer Center, etc. With that, I rest my case, and now I'll be more than happy to answer any question you may have. How to remove and Brenda?
Just
Escape, right? No. Can I say? Yeah, so. Yeah, so can we make the. You have any question, or do you think I ran out of time? Or?
Yeah, we can have quick questions if anybody have.
I tried to make.
Okay.
Yes, please,
Dr. Mahato, I have a quick question.
Yeah, stop saying. Yeah, please tell me. Yeah.
Yeah, it was wonderful talk. I'm very happy that I could listen. And so just one quick question about technical things, that is it always required to prove the synergism by some techniques like statistical techniques like Chow-Thalai methods or something, or just cell viability like significant inhibition of the cells, proliferative pathways are enough?
No, actually, you have to do many different ways to prove there is a synergistic effect or therapeutic effect. So I just showed you one cell viability, but you have to also do like different marker like colony formation assay and so on. So there's a lot of technique you have to do, but in the presentation, naturally, I can't show you all because I just simply wanted to make a story, but you have to do all other technique. But other really, I want for you to think of, rather than killing a lot of times in vitro dealing with cells, I would suggest to really go in animal model and the right animal model, so that you will have better impact of your study. Did I answer your question or not? Yeah.
Yeah, go ahead, sir.
So it means you have to do more than one technique, like mRNA level, like Western blot level, immunohistochemistry, and also you have to use a statistical analysis. So most of we do apply statistical analysis, but honestly, now I reached the phase I don't have time to prepare those things, so I just do it very fast. But people in the lab, they certainly apply all the statistics because this is absolute requirement. Because as you know, NIH now is very serious about reproducibility of the data, and that's why statistical analysis become very, very important.
Right. Okay. Thank you. Thank you.
Yeah. Any other question?
Professor Mahato, I have a quick question. Whenever we are talking about combination therapy, generally we target the same cancer cell, right? From different mechanism. Now, what is your suggestion regarding finding out those targets? That this pathway I can target along with that one. So
my suggestion is, number one, don't use the two different combinations for the same target. Even if you use the same disease, tumor or maybe other disease, right? but that can be, I give you example, like for example, like pancreatic cancer, right? So one can be chemo resistance, but can be also hedgehog issue, right? Can be dysplasia, right? So if you use more than a couple of target, then the benefit will be better, So that's, and another thing is try to target not only bulk tumor, but quite often tumor initiating cells. Because that's very, very important. But in some cases, for example, if you do like liver, like cancer, in that case also inflammation become very important. So depending on the disease types, you may have to choose wisely. And that's why cancer biology or any disease pathophysiology understanding is super very, very important. And that's why I, especially in my lab, I require a student to really invest some time to understand cancer biology or if other disease pathways, so that they should know different signaling pathway, and that way they will select the right pathway, combination therapy, because you don't want additive effect, right? You want synergistic effect, right? Yeah.
Synergistic effect. Thank you so much, sir. Thank you.
Yeah. You're welcome. Yeah. If any other question, if not, then you are more than welcome to write to me. I will be happy to answer any question if you have, and certainly it was my pleasure to talking to you all. And Deepak, anything else you want? If not, then
thank you. Thank you, Professor Mark, for giving this insightful talk and motivating the students and young faculties like us to look deeper into the field. Thank you for spending your time for that talk.
Yeah, that's most important, because especially for young faculty, I'm not old, but still I feel a little bit older than some of you. If you're younger faculty, don't go for quick return, you invest something to go deeper, and that's very important. That's something I made a mistake, because I tried to have a quick return, and in the process I become very superficial. So I would strongly recommend to go a little bit deeper, and publish in a high impact journal, which again, most of us, including myself, are failing to do so. Yeah, thank you.
Yeah, thank you, Dr. Mark.
Thank you.
So, Professor Lee, Professor Shetali, are you there?
Yeah, I'm here.
Yeah. So our next speaker is Professor Shardar Lee from University of British Columbia. So let me introduce Professor Lee to the audience. So Dr. Lee has received his BSc in Pharmacy from National Taiwan University in 1998 and PhD in Pharmaceutical Sciences from the University of North Carolina at Chapel Hill in 2008. He finished his postdoctoral training at Morris Cancer Center at the University of California, San Diego in 2009. He is now an associate professor at UBC Pharmaceutical Sciences. Dr. Lee has won several research awards, including 2014 AFPC New Investigator Award, 2013 APS New Investigator Award in Pharmaceutics and Pharmaceutical Technologies, 2013 CIHR New Investigator Award, 2013 CSPS Early Career Award, and many more. He is the chair-elect of the Targeted Drug Delivery and Product Focus Group at APS and an academic editor of Biomed Research International. Dr. Lee's research focuses on developing innovative drug delivery systems to enhance cancer chemotherapy. His research program has been well supported by the NIH and CIHR. So with this, I invite Professor Lee for the talk. Now it's over to you, Professor Lee.
Okay, thank you so much. Can you see my screen?
Yeah, not yet. Can you please share that?
I think I did. Is it good now?
Professor Lee, you probably have to go to the right side, low corner in the screen. There is a thing like present now. If you click there, then you can share your window or your entire screen.
Okay, let's see. I see. Okay. So give me entire screen. Can you see my screen now?
Yes, it is
Yes, we
visible.
Okay, great. Thanks so much for the kind introduction and the invitation for me to share my work with you today. So today I'm just going to share a little bit about my research program at University of British Columbia in Canada. So at UBC, we are working on drug delivery technologies for targeted therapies. And we are working on many different technologies, including nanoparticles and prodrugs. And today I just want to talk about one of our focuses, which is liposomes. So many of you are very familiar with liposomes. So the structure of liposomes is shown here. Basically, is as you can see, this is a liposome. The structure is basically a lipid bilayer. And for drug delivery purposes, we actually make these liposomes in the nano size. So you can view liposomes as a nano-sized cell. So it's able to deliver both hydrophobic and hydrophilic drugs. But in our lab, we are mainly focused on polywater-soluble drugs. And today I want to discuss two research topics we are working on with liposomes. The first one is that how do we change lipid composition or the fabrication way to make liposomes targeted to different disease sites. And today I'm going to focus on hepatocytes. And second part, as I mentioned, we are interested in targeted delivery of polywater-soluble drugs. But what we are working on is actually developing a very unique active loading technology to load those polywater-soluble drugs into the aqueous core of liposomes, so that we are enhancing the stability of the drug during the circulation, and also using nanoparticle lipid nanoparticles, in this case liposomes, to target these drugs to the disease sites. Again, many of you are familiar with the manufacturing of liposomes. So it all started with a thin lipid film, and with hydration, then the lipids will self-assemble into so-called multilamellar vesicles. And we can further extrude MLV into SUV, stands for small unilaminar vesicles. And by just passing them through a very precise membrane with precise pore size. So this is how we create SUV or liposomes for a targeted drug delivery. And in terms of drug loading, right now most people are using the so-called active loading drug technology. So first they create a transmembrane gradient that will allow drug to participate into the inner aqueous core. But definitely there are some limitations for this thin film hydration method and active targeting method. And this is what our lab is currently working on to address those limitations. First, I want to talk about a new project in the lab. Is phospholipid-free small unilamellar vesicle, or so-called PFSUV. So as I mentioned, for liposome or liposomal SUV, they are composed of phospholipids. But we also know that SUVs can form with just cholesterol and non-ionic surfactant at 1:1 molar ratio. But this standard so-called neosome formulation is actually not very stable in the plasma because it has to contain a very high non-ionic surfactant component in the formulation to help cholesterol disperse in this good lipid bilayer structure. So that creates a lot of problem because of poor stability. Then the formulation is mainly used for topical application, not for systemic application. So here at UBC, we are using this very innovative microfluidic system called herringbone microfluidics. So it allows organic solvents to collide with aqueous buffer in a very efficient and very rapid way. So in this case, we can dissolve lipids or other poorly water-soluble components in an organic solvent and mix with an aqueous buffer within this microfluidics to produce our nanoparticles. So in this case, we actually just used cholesterol and 2080, which is one of the non-ionic surfactants that are commonly used for drug delivery. So our goal was to increase cholesterol content as high as possible, and we were able to achieve a 5-to-1 molar ratio of cholesterol to 2080. So this, as you can see, with this technology or fabrication method, we were able to produce SUV contains no phospholipids, but only cholesterol and 2080 in a very efficient way. And these SUVs, as you can see, they contain a very nice lipid bilayer, and the size is about 50 to 60 nanometer, up to 80 nanometer. So pretty small. And we were wondering whether these nanoparticles can be loaded with drugs using active loading technology. So we use a weak-based drug as a model drug. And so as you can see that when we create this pH gradient, we were able to drive this weak-based drug, which is doxorubicin, into the aqueous core of the PFSUVs. So these doxorubicin actually form crystals inside the SUVs. You can see this nice structure inside. The next thing we tested is the stability of PFSUVs in plasma. As I mentioned earlier, the standard neosol formulation, because they contain a lot of non-ionic surfactant, that makes them very unstable in plasma. But in this case, we look at the drug retention in the serum and then compare two different formulations. One is PFSUV, the other is pegylated liposomal doxorubicin, which is the equivalent of Doxil or Calyx, currently used in the clinic for breast cancer therapy. And you can see that they are almost as stable. You see that within 3 days, there's no drug leakage from PFSUV in the presence of serum. And even at day 6, we still maintain 90% of the drug content inside the PFSUV. So quite stable. Then this makes it feasible for this formulation to be administered through a systemic route. So this is a in vivo imaging study. We label PFSUVs with a fluorescent dye and then injected these fluorescently labeled PFSUVs into mice. And over time, we just image those mice. And it's not really surprising to see a very rapid accumulation in liver. But I have to say that the tissue targeting is pretty specific to the liver. And what is really interesting is that when we look into the liver section and we discover that these nanoparticles are actually targeting the hepatocytes rather than the Kupffer cells, like most of other nanoparticles reported in the literature. So in here, this is a liver section. And we scan the outline, the skeleton of the the cells with phalloidin, so you can see the cell outline pretty well in green, and our nanoparticles are labeled in red. And with this, you can clearly differentiate the Kupffer cells from hepatocytes by the green outline and the nuclear morphology. So for example, this is a Kupffer cell, but this big structure is a hepatocyte. And you can pretty much appreciate that most of the red signal are within the hepatocytes. And you see some of the Kupffer cells have some uptake, but for example, this one doesn't really have any red signal. But in here, most of the hepatocytes are stained red. So it's suggesting very selective targeting for hepatocytes for our PFSUVs. So we did some quantitative analysis looking at DIR, which is the fluorescent dye positive cells per field, and you see hepatocyte or the major cell type. On the other hand, we also look at within each cell type, do we have a higher uptake? As you can see here, only among all the hepatocytes, 70% of them are stained with PFSUVs, but only 30% of the Kupffer cells are positive with our PFSUVs, again suggesting this is a really hepatocyte selective delivery system. So we have performed some preliminary study to look at the mechanism of this PFSUV for hepatocyte targeting. And our preliminary data suggested that these nanoparticles actually, when they are injected into the blood circulation, they absorb apolipoproteins to their surface. And because of the small size, they were able to penetrate through the liver fenestra and then into the space that can interact with the hepatocytes through the LDL receptor pathway. And the following just that we did some work just to show that these very unique PFSUV particles are able to target drugs to have some medical applications. So in this case, we are looking at acetaminophen-induced liver injury. So this is still the number one cause for acute liver injury in the Western world. So the model is induced by IP injection of 500 milligram per kilogram APAP, which is acetaminophen. And 1 hour later, we injected a drug called chlorpromazine, which is an autophagy inducer that has been shown to rescue liver or hepatocytes after acetaminophen acute tox. And 6 hours later, we collect the plasma and also the liver to examine the liver toxicity. So as you can see here, the untreated liver shows a big region of necrosis. With some free drug treatment, you were able to see reduction of the necrotic area, but the necrosis is still significant. With our PFSUV, the liver histology looks almost normal. And we also compare ALT, which is the liver enzyme that is reduced after liver is injured. Again, we can see that with PFSUV treatment, the serum ALT concentration is much reduced compared to untreated control and free drug. So the other application we look at is liver stage malaria. So malaria is still the most prevalent parasitic diseases in the world. So it causes a lot of death each year. We do have a lot of drugs that are very effective against the circulating parasites of the malaria. But the problem is those bugs actually hide in the liver, and so those drugs are no longer effective against the liver stage malaria, and those parasites can relapse anytime. So it becomes very difficult to cure malaria in this case. But we do have at least one drug that is effective for both circulating and liver stage malaria. One of them is primaquine. The structure is shown here is a wheat-based drug. The problem is that this drug is inducing very significant hemolysis. So this significant toxicity prevents its wide use in older patients. So we were wondering maybe our PFS-UV can target this primaquine, this PQ drug, to the hepatocytes more effectively. But in the meantime, during the circulation, the particles were able to protect the drug from entering into the RBCs to reduce the hemolysis toxicity. So indeed, when we did the animal study, when we examined the liver uptake, we were able to see a very significantly increased the liver uptake with our PFS-UV formulation compared to free PQ by oral or IV. So especially for oral free PQ, our PFS-UV increased the liver delivery by 5-fold. On the other hand, the RBC uptake was reduced by 15-fold, so very significant. And we also look at whether PFS-UV were able to decrease the RBC toxicity. So this is looking at the RBC morphology after different treatments. So this is a very nice RBC round shape, very nice cell membrane still. But with free PQ treatment, you can see that the membrane is sort of deformed a little bit, suggesting this might be toxicity there. And with the PFS-UV, you see the morphology of RBC is preserved better compared to free PQ treatment. So that concludes the part one, which we use a unique fabrication way, and then we clear the lipid composition for a specific cell or tissue targeting. The second part of my talk will be focused on so-called active loading technology. So this is the standard active drug loading technology for liposomes. So as I mentioned, first you create a gradient, a transmembrane gradient between the liposomes. And you just add an antipathic drug in the external media. And because the drug is soluble, so it will be soluble in the exterior buffer. But in the meantime, it's also highly membrane permeable. So it will permeate through the lipid bilayer and then interact with the internal gradient. In this case, if you put a trapping agent inside, it will form complexes with this trapping agent and get locked inside the liposomes. So this is the standard active loading technology. But it provides several advantages. For example, the drug loading capacity and efficiency is much higher with active loading compared to the other method called passive loading. And because the drug is stably loaded inside, the in vivo stability is increased. So these are the two major advantages. But the limitation is that this technology only applies for homeopathic drugs, meaning the drug needs to have good solubility, a good membrane permeability to be able to actively load it into liposomes by this technology. But as we know that most of the investigational drugs are poorly water soluble. So that's what we are focused on, developing this so-called solvent-assisted active loading technology. In short, SALT for poorly water soluble drugs. So again, our idea is that first we still create a liposome formulation containing a transmembrane gradient. But before we add our drug, we actually included a small amount of water-miscible solvent in this liposome formulation. And when we add this poorly water-soluble, then the solvent will have two functions. First, it will increase the solubility of the drug in the mixture. Second, it will increase the membrane permeability, allowing the drug to penetrate into the inner aqueous core to form complexes with the trapping agent. At the end, we just perform dialysis to remove the solvent or any unencapsulated drug to obtain the final purified liposomal drug formulation. So that was just a concept. So the first thing we did to demonstrate this is a feasible method is using a model drug to test our hypothesis. So the drug is called staurosporine with a structure shown here. Although it's a weak-based drug, the drug is actually not soluble in water. It's only soluble in DMSO. So our approach is that we create this ammonium sulfate gradient for weak-based loading. And then we compare either no DMSO in the loading mixture or the loading mixture contains 5% DMSO, and we compare the drug loading efficiency. As you can see, without DMSO, all the drug is precipitated outside the exterior phase without significant drug loading. But with 5%, we were able to load all the drug molecules inside the liposomes. And so the cryo-TM, just to demonstrate that indeed the drug is loaded inside the aqueous core rather than a bilayer, like most other reports have demonstrated. So as you can see that you can see drug crystals inside the PFS-UV particles, liposomal particles. Okay, let's look into the applications of this SALT technology. So the first example is a Chinese medicine. So as you know that a lot of active compounds have been isolated from traditional Chinese medicine or herbal medicine. But they do have significant problems. First, most of them are actually not water soluble. So one example is gambagic acid. The structure is shown here is a weak acid drug, but most of the structure is very hydrophobic. So as a result, gambagic acid is not soluble in water. But it has been shown to have several really exciting biological functions or pharmacological activities. So including anti-inflammatory, anti-angiogenesis, and anti-proliferation. So this drug has been heavily investigated for cancer therapy. But again, the water solubility is one of the limitations. So in here, we are using this copper acetate as a trapping agent for this model drug. And we were able to show that we could use many different solvents in the loading mixture, containing just 5 to 7% water miscible solvents of this list of solvents to actively load gambagic acid into the liposomes. So I just want to show you that this is our final product of LipoGA. The size is about 18 nanometer, and the drug to lipid ratio is 1:5 weight ratio, so very high drug loading. And as I mentioned, the problem with gambagic acid is poor solubility. So we were able to increase the apparent solubility from 8 microgram per ml to greater than 10 milligram per ml. So very significant improvement in terms of solubility. So what is the working mechanism for salt? So what we have done is a very systemic analysis between the loading efficiency and the percentage of miscible solvent within the loading mixture. And what we have observed is that there is definitely a optimal solvent range for complete drug loading. Okay, so below that is not good, but exceed that range is definitely not good either. And another thing is that we also discovered that if we want to achieve complete drug loading, we need to add a little bit more solvent than the amount that is required to completely dissolve the drug. So the results suggested that the solvent is acting through two different mechanisms. First, definitely to dissolve the drug, but just completely dissolving the drug is not enough because you need slightly above that. So we are anticipating that maybe the slightly over solvent requirement is to increase the membrane permeability to allow the drug to penetrate through the bilayer more effectively. And also, if you include too much solvent, then the membrane might become unstable and the gradient might collapse to prevent drug loading. So this is the working mechanism for salt. Let's go back to the gambagic acid story. So poor water solubility is one of the limitations. Another limitation is that GA induces significant hemolytic toxicity. So in here, we are comparing the hemolysis of free GA with our LipoGA. As you can see, free GA is very toxic to RBC. But after liposomal encapsulation, the drug becomes safe. So we also look into the anti-tumor efficacy. We look at multiple animal model or tumor models. Maybe I will just focus on the EMT6 breast cancer on the left-hand side. As you can see, the tumor is quite aggressive. They grow very fast. With the free GA treatment shown in green line here, shown in the orange line here, there's no significant effect. Probably because of the dose we could safely inject it was not effective. But if we load the drug into liposomes, even at a low dose, we were able to show significant results. And because of the reduced toxicity, we could even inject a much higher dose at 20 milligram per kilogram, and we were able to completely suppress the tumor growth. So this is another model showing comparable results. And I also want to introduce a second application, which is quite interesting in my opinion, that we use this technology to help us prepare child-friendly oral formulation. As you know, that pediatric formulation is not a lot of people working on this field because first, it's very challenging. Second, the market is small. But I think this is a very important topic to work on, especially for academic scientists. So what are the requirements for pediatric formulations though? First, liquid over solid, because most children have a swallowing problem. And the taste is very important. And only safe ingredients and bioavailable. And I was thinking, well, maybe our liposomal formulation will serve the purpose, right? Because we could load drug inside the particles, and we could, in that way, we could increase the solubility to prepare a liquid formulation. And also, the drug molecule is now loaded inside the bilayer, and the bilayer will prevent direct contact between the drug molecules with the taste buds. And this would be an effective way to mask the taste of many drugs. And again, I'm using malaria drug as a model drug. So I'm going to skip the introduction, just go into the results. So the model drug is mefloquine. It's super bitter. It's still the first line drug for malaria in Africa, but it doesn't really have an pediatric formulation. So in those countries, what has been done is that pharmacists will just take adult tablets of mefloquine and break it into pieces and crushed and into powder, and then add to water or liquid or food and feed to the children. But as you know, that is not going to mask the taste well, so the medication adherence is not good. So many children just spit out. That affected the accurate dosing and the medication adherence. So we use our technology, the salt technology, to load mefloquine, which is a poorly water-soluble drug, as well into the liposomes. And the first thing we look at is the taste. So we measure the bitterness by the e-tongue technology, basically a biosensor that is able to quantify bitterness. And as you see here, we use 10% sucrose as a reference. It shows negative results, suggesting it's sweet. And mefloquine suspension, as you know, is right now used in Africa, very bitter. And we have infant Tylenol as a reference too, just to show that what is the acceptable range of bitterness, which in this case is 250. But look at mefloquine liposome, is it tastes like a sugar water here. The next thing we look at is looking at whether our formulation can release mefloquine in simulated gastrointestinal fluids. And here is simulated saliva. We don't want the drug to release in the saliva. So again, no drug release. And in here we have mefloquine suspension, very rapid release, so causing bitterness in the mouth. And we have two mefloquine liposome formulation. The only difference is just a different drug to lipid ratio. So you see no drug release. But in the simulated gastric fluid, you see rapid release for all formulations. And in the simulated intestinal fluid, we see rapid release as well. So the data suggests that our mefloquine liposomal formulation will not release in the oral cavity to induce bitterness, but will be rapidly released in the gastrointestinal conditions to allow the rapid drug absorption. And finally, we look at the oral absorption in a mouse model. So just very quickly, you see that our liposomal mefloquine shown in purple line here is absorbed very rapidly, producing a very good blood profile in mice. It's comparable as mefloquine suspension, or even a little bit better because it increases solubility. So another example I want to show is lyophilized mefloquine liposome. So we know that liquid formulation is difficult to store. So the lyophilized mefloquine liposomal formulation is to resolve that challenge by drying the liquid formulation into powder. But this powder formulation is able to be reconstituted in just a second. And we show that with this lyophilized formulation, we were still able to produce very good blood profile. And we also confirmed that after lyophilization, mefloquine drug molecules is still loaded inside the liposomes. And the final part of my talk today is, again, using this salt technology as to prepare a immunotherapy formulation for treating intraperitoneal metastasis of cancer. So we know that intraperitoneal metastases are often found with ovarian cancer, pancreatic cancer, and colon cancer. They are very difficult to treat. And one of the characteristics of these metastases is that the tumor microenvironment is highly immunosuppressed. So we were wondering whether we could use a drug delivery technology to wake up the immune system in the peritoneum cavity to promote anti-tumor immunity. So our approach is that we want to use an immune boosting agent, and we want to localize this immune boosting agent in the peritoneal cavity. So the immune boosting agent we are using is RA4A, is a toll-like receptor 7A agonist. So it's very effective in activating dendritic cells to secrete interferon alpha to promote anti-tumor immunity. But the problem with RA4A is, first, is the water is poorly water soluble. Second is that if it's injected and distributed throughout the body, then it will induce non-specific immune response globally or systemically to induce very significant side effects. So the delivery must be localized to the peritoneal cavity to reduce the toxicity and improve the efficacy. So again, we were thinking to use our salt technology to load these drugs into liposomes and use liposome to localize this drug in the peritoneal cavity. The first thing we did was looking at how different formulation of liposomes would affect the peritoneal retention. We compare the charge effect. So we prepare negative, neutral, and positive formulations of liposomes, and then IP deliver these formulations into mice and look at their IP retention. As you can see that the positively charged liposomes are very effective in retaining in the peritoneum compared to the negative and neutral liposomes. So especially for negative formulations, you see a very rapid absorption after 1 hour and to the rest of the body. So then we quickly look at the pharmacokinetics and the biodistribution of the formulation in comparison with free drug in these components, including peritoneal fluid, IP cells, which is cells collected from peritoneal fluid, and then plasma. So what we hope to achieve is that liposomes or DS10 liposomes was able to increase the peritoneal fluid concentration, increase the delivery to IP cells, but reduced absorption or slower absorption into plasma. And you see that we achieve these as shown that the red data representing DS-TAB RA4A are much higher in the peritoneal fluid and IP cells, but the absorption to the systemic to the system is much delayed compared to the free RA4A. And we were also wondering within those IP cells, what kind of cells are we deliver our DS-TAB RA4A to. So we label the DS-TAB liposomes with a fluorescent dye and IP injected into mice and examine those Di positive or Di labeled liposomes and Di positive cell population. So within all the Di positive cells in the IP in the peritoneal cavity, a lot of them, like 50% of them, are monocytes, and about 10% are dendritic cells, 10% are NK cells. So this is to our expectation. Okay, but we also look at within each cell population, how many of them are positive with the Di. For example, monocytes here. Actually, among all the monocytes in the peritoneal cavity, only 0.5% are positive. So actually, in this, we can conclude that monocytes are not selectively targeted by our formulation because only 0.5% of them are positive. The reason that we see a lot of Di positive cells are monocytes is because monocytes are the vast majority population in the peritoneal cavity. But what is really interesting is the dendritic cells. We see very, very high percentage of the dendritic cells are positive with Di, about 40%. And this mature macrophages are very effective. Are very effectively targeted as well, but their population in the peritoneal cavity is pretty small. So the results suggest that the major immune cell population that we are targeting to is dendritic cells, which is what we hope to target. As I mentioned, interferon alpha is the major cytokine induced by the RA4A activation, and also the very important cytokine that will turn a cold tumor into a hot tumor. So this is the cytokine that we look at first. So as you can see here, we look at the mRNA expression from these IP cells. And you see that with DS-TAB treatment, we were able to increase the interferon alpha expression by 10-fold compared to the control, while free RA4A was less effective, only inducing 5-fold. And finally, we look at the efficacy because all the data are very positive in supporting that we are inducing a very good immune response. So in here, we are introducing CD26 colon cancer into mice by IP injection. We also label these cells with a luciferase tag so that we can monitor the tumor progression non-invasively using bioluminescence imaging. And on day one, we inject oxaliplatin, which is commonly used anti-cancer drug for colon cancer. And the next day, we IP inject either PBS, free RA4A, or DS-TAB RA4A. And then we monitor tumor progression using bioluminescence imaging. As you see that these tumors actually grow really fast. So on day 13, the tumor burden are really significant. But with oxaliplatin DS-TAP combination, we were able to slow down the progression. And here we show the quantitative results. And in here, the white bar shows oxaliplatin plus PBS. There's no control of the tumor progression. With combination of free RFOA, you see initial response on day 3 and day 4, but tumor quickly rebound and uncontrolled. And so, as I have shown earlier, free RFOA was able to induce interferon alpha release. So, this early effect probably from non-specific effect from interferon alpha. With the combination with DS-TAP, the effect is more prolonged. Even on day 13, it shows significant tumor inhibition compared to PBS and the free RFOA group. So our data suggests that there might be some immune response against the tumor. So another thing we look at is whether our treatment has changed the immune microenvironment. So in here, we isolate the peritoneal fluid and then look at the T cell and differentiate them between the CD4 and CD8 population. So in the first group is oxaliplatin plus PBS. You see that the CD8 population is 76% among all the T cells, and CD4 population is about 16%. And after combination with RA4A, you see the CD8 population increases to 82%, and CD4 population decreases to 10%. With DS-TAP RFOA treatments, the CD8 positive population further increased to 93%, and CD4 population further decreased to 3%. So for the anti-cancer therapy, CD8 population is preferred because they are cytotoxic T cells. They were able to recognize cancer cells and then perform lysis. So we believe the mechanism is preferred for significant cytotoxic anti-cancer effect from the immune response. So this slide just summarizes our proposed mechanism supported by our data. We believe that oxaliplatin is first induced double-strand DNA breaks, inducing so-called immunogenic cell death, and the released antigen together with DS-TAP RFOA delivery to the DCs will activate the dendritic cells to the active part, to the active counterpart, and then the activated DCs will secrete interferon alpha, and then they will interact with T cells and then activate T cells into CD8 positive T cells. And these CD8 cells will then perform tumor lysis to suppress the tumor. So I just want to quickly summarize my talk today. So again, we focused on formulation and the manufacturing method development for liposomal drug delivery systems. And we use these technologies to target drugs to specific sites. And today I give you one example for hepatocyte targeting. And second, we are interested in drug loading technologies, especially for poorly water-soluble drugs. And today I show you several examples of how we can use this solvent-assisted active loading technology to load a variety of drugs to improve their targeting. I just want to acknowledge my lab and my collaborators and funding. Thank you so much for listening.
So thank you, Professor Lee. So we can have a couple of quick questions if anybody have.
Thank you, Deepak. I might ask a question.
Yes, Professor.
Professor Lee, very nice presentation. I learned a lot. I had a question about your the salt loading technology. When you're using solvents like DMSO or DMF, how difficult it is to remove them after you formulate them? And was it done by dialysis or freeze drying? How did you do that?
Yeah, so far we were using dialysis to remove those residual solvents, and we monitor residual solvents throughout the dialysis process. And what we found is that after 2 hours, those solvents are under detection limits. But we actually performed dialysis much longer than 2 hours. And we did multiple changes of the solvent, I mean the buffer.
Sure. And then the other quick question was that these lipids that you're using, are they able to survive the lipases and the gastric environment? Are they lipids that are specifically chosen for oral delivery?
Yeah, so I think you were referring to the pediatric formulation part. So we did see some very rapid release in the simulated intestinal fluid. And so we actually examined the mechanism. So first we were wondering whether it's through the lipase digestion, but it's actually not, because when we incubate our liposomes with the lipase that we purchased from Sigma, and we incubate them for several hours, and we didn't see any degradation. So the release mechanism is actually through the sodium tauchlate or the bile salt that we supplemented into the simulated fluid. Because in the normal intestinal conditions, there will be some bile salts presenting in the intestinal fluid. So that's what we have found to be the mechanism.
Okay, excellent. Thank you so much.
Yep.
Sir, there is one question. Like you have talked about the amphiphilic drugs. So what are the properties or the characterization we can do for these drugs, and how the nanocarriers should have some property or groups on it so that it can be loaded efficiently inside the formulation?
Yeah, so if you are talking about the salt technology for loading drugs into the aqueous core of liposomes, we do have some criteria for selecting those drugs. Definitely, you need to have a good loading gradient and so-called trapping agent inside the liposomes to interact with your drug, right? So the most successful example we have obtained so far are wheat-based drugs that containing amino groups that can form complexes with sulfate ion. But we were also successful with the carboxylate functional groups, and we were use copper gradient or copper to complex with those functional groups. So I think the functional groups presenting in the drug you are hoping to load is very important. That will decide what kind of gradient you need to use. But in terms of our limitations, because our technology is trying to resolve the issue of or the limitation of current active loading technology that require drug to be both soluble in water and membrane permeable. right? So I think I have shown you several examples that as long as the drug is, has some functional group that you can utilize, there's a high chance that our active loading technology will work for them.
Okay. Sir, another small question is, if the drug is low water soluble and low low lipophilicity is there, then what will be the liposomal fate according to the formulation?
Right. So I would say most those are low solubility and poor membrane permeability drugs. They are because of they are a substrate for PGP, right? So that they are characterized in that category. But in our liposomal delivery, because there's no PGP there, so normally if the chemical structure shows that the permeability is okay, then it should be able to load.
Okay, thank you, sir. Thank you.
Yep.
Yeah, thank you, Professor Lee, for providing the insights on the phospholipid-free SUVs for targeting and discussing the active loading technology with the audience. So I thank you for sparing your time for this talk. Thank you, Professor Lee.
Thank you. Yeah, thank you.
So now our next speaker is Professor Amirali Popat from University of Queensland, Australia. So Associate Professor Amirali Popat is a NHMRC Fellow and Director of the Research at the University of Queensland School of Pharmacy. He has a PhD from Australian Institute of Bioengineering and Nanotechnology in 2012. Dr. Popat has a Bachelor of Pharmacy from KBI-PER and Master of Pharmaceutics from LMCP, and has research interest in advanced drug delivery and nanomedicine. He has published over 45 journal articles and have 3 patents in his name. His group has received more than $6 million of funding in the past 5 years. He is also the current president of Australian Control Release Society and academic board member of the CRS focus group on nanoscale delivery. He is also an advisory board member of many journals such as Pharmaceutics, DDTR, and Nanoscale Horizon. So with this, I invite Professor Amir to give his talk. Over to you, Professor Amir.
Thank you. Thank you, Deepak, and thank you to Professor Jadhav for the kind invitation. Are you able to see my whole screen?
Yes, yeah, you can put it on the full view mode.
Okay, all right. Okay, this is visible, right?
Yes.
Okay, great. Thank you all for joining for this excellent conference that has been so far. Today, what I'll talk a little bit about is one of the technologies that we are using currently in our lab. My lab is situated at the School of Pharmacy, University of Queensland, Australia. And we particularly are interested in development of new materials or synthesizing new materials for drug delivery. We do work with traditional liposomes, PLGA type materials, but I think the major focus of the lab has been for the past 5 to 7 years is to develop a new type of material. And one of those new type of material is silica-based nanoformulations, and I'm going to talk a little bit more about that. So before I start, I really like this this paper from Robert Langers in Journal of American Chemical Society, JACS, in 2016, sort of re-highlighting the need of how traditionally the drug delivery technologies were made and what is the role of the new entities, new chemical entities, or the biologicals, or those interferon alpha type molecules, immunotherapeutic drugs, and how the drug delivery has sort of evolved. This is a quite traditional graph BCS classification, and majority of you are aware of this classification, which sort of talks about poor permeability, poor solubility of small molecules, and not more than 90% of those newly synthesized molecules are either poorly soluble or poorly permeable or both. However, only less than 1% of the biologics can be delivered orally. And even that 1% of biologics that can be delivered orally, the bioavailability is very, very poor. So again, if we think about the traditionally how the drugs were formulated is using this controlled release technologies such as reservoir type polymers, degradable materials, erodible materials, osmotic pump, and hydrogels. When osmotic pump and hydrogels came into the market, it was a very big thing, and it sort of solved problems of the drugs at the time very rapidly. Hydrogels were then also popular in wound healing and other application as well. But this article from Robert Langer also talks about the evolving role of drug delivery in a way that the new molecules that are being synthesized, the biologics that are being developed, peptides, proteins, immunotherapeutic agents, how do we deliver them? So it talks about thinking outside the box and thinking about new materials rather than keep using existing materials. Certainly one way to do that is modifying or improving on existing materials. And, Professor Lee has given a very important example of liposomes, and the drug loading in the liposome has been an issue, and how they've been able to solve that problem. So that's excellent in terms of getting that into the market, because liposomes are, there's a history there, and majority of the nanomedicine product are liposome-based products that are in the market for cancer. So when we started designing those materials, we wanted to think about the challenges that there are already there and what is out there. So first of all, if we think about the challenges are your salivary enzymes. As soon as you take the tablet orally or capsule orally or patch or oral dispersible formulations, and then it goes to the stomach where there's pepsine, and the pH is really, pH is a problem as well. And then when the formulations are going to colon, those formulations are then degraded by your enzymes, and then permeability also an issue. If you focus here on the mucus barrier, so after the formulation has passed the stomach, in the small intestine, mucus barrier is a major barrier, and that mucus barrier not only stops the bacteria, then pathogens to enter the body, so that's really great, but also stops the molecules and some of the drugs that are not permeable, and sort of stops the prevent them to be orally available. So various strategies have been designed that you have mucus penetrating nanoparticles. So you put hydrophilic polymers such as, polyethylene glycol or polyethylene glycol derivatives. You also have mucoadhesive patches that you could use. So mucoadhesive has also been another approach where you use chitosan or positively charged polymers. Enzyme inhibition is, very interestingly used by this orally available semaglutide formulation that has been recently approved. That technology uses a small molecule enzyme inhibitor called SNAC, and that actually increases the pH in the stomach and allows the semaglutide to be absorbed while inactivating the enzyme that actually degrades it. So that's another strategy that has been used to improve the oral delivery. Paracellular transport, so opening the tight junction in the gut transiently and allowing the molecule to pass through. One of the challenges there might be that when you open the tight junction, other pathogens might also attack the body. But if you carefully do that, that's also another interesting strategy. Transcellular transport is an active transport mechanism where your carrier actually takes your drug through into the epithelial cell via endocytosis and then exocytosis into the bloodstream. And in terms of the skin-based delivery, or taking molecules through the skin into the bloodstream, microneedles have been also used. However, if you look at these traditionally used systems, there are several disadvantages of these systems, and one of the disadvantages is, premature drug release. And a typical example of that is PLGA. PLGA has been used for many decades now. However, still the loading capacity and the premature drug release are the two major issues. Another issue is that a lot of these polymers are also prone to solvent degradation. So if you want to modify the PLG and nanoparticles with several different functionalities, it is very difficult to do that unless you design those functionality into the monomers, or it is certainly possible slightly more with liposomes, but with PLG and or type polymer nanoparticles, very difficult. Some of the lipid-based formulation are prone to acid degradation. Some of the lipids that Professor Lee mentioned are not, so there is an improvement there. Again, low drug loading capacity of the liposome is an issue which Dr. Lee and their labs have been able to tackle. But burst release. So if you look at the release profile from the liposomes, it is very rapid. The liposomes or the lipid-based SMED type formulations are able to improve the solubility. They are able to improve the dissolution. But as soon as you put them into the media, or as soon as they are in the gut, they will solubilize the drug. Now, that's not a problem. Sometimes you want a drug to be released in a controlled manner. Other times, the drug that is completely solubilized may also start to reprecipitate. So you see a burst biological effect, and that is followed by crystallization of the drug inside the body because it's dumped as in the body that way, and that can be a challenge. And solid particles such as gold, silver, they have a great use in diagnosis. They're great use in as an antimicrobial agents. But when you talk about delivering antimalarial drug, antibiotics, or anti-inflammatory drugs, these nanoparticles are not that great because the amount of nanoparticle you require to be given to the patient will exceed the toxicity limit of those. So there's a need to formulate bioresponsive nanoparticle which possess high drug loading capacity. So I'm talking between 10 and 25% by weight. I'm not talking about 25% of the encapsulation efficiency, but I'm talking about 25% by weight. So 1 gram of my formulation should contain 200 to 250 milligram of active drug. That can improve the solubility. You can tailor them to control the release of the drug. They should be biodegradable like liposomes and PLGA, and that should provide site-specific oral delivery. Fortunately, during my PhD in 2011 and 12, what we were working with is mesoporous silica nanoparticles. Now, these nanoparticles are inorganic nanoparticles discovered by Mobil, oil company, and that was mainly discovered for catalysis. They are very interesting candidates because they have very high surface area. The particle size can be tuned, pore size can be tuned. And the use of silica in tableting industry is not new. It has been used, aerosol has been used as a tableting excipients since many, many decades. Now, the figure on the right actually tells you that how easy it is to functionalize them, put different drugs, put the antibody. You can functionalize them with quantum dots, iron oxide nanoparticles, peptides, polymers, lipids. Surface functionalization, you can load plasmid DNA, you can encapsulate siRNA, you can put aptamer, and then particles can actually cross the many membranes and barriers as well. So how do we synthesize them? So for those of you who are working in the oral drug delivery or cancer-based nanomedicine, synthesis of silica nanoparticles is actually very, very simple. What you need to think about a fundamental chemistry. So when you have a surfactant, all surfactants have critical micelle concentration. So at the critical micellar concentration, those surfactants will self-assemble. And once they self-assemble, the shape of the micelle, the size of the micelle will determine the final morphology. But what you can do is you can change that by changing the parameters such as pH, the amount of surfactant, the type of surfactant, a solvent, and then you add a different types of silica source that can have an impact on the morphology. So once you add the silica source such as the triethylorthosilicate or its derivatives, it will start to react with those surfactants on the surface, and then it will polymerize and condense to form this thick wall outside those surfactants. Now, if you remove the surfactant or selectively etch the surfactant, what you will get is actually a pores. And now those pores are not pores that are physically done. They are done at a molecular level, and the size and the shape of the pore is exactly the same for the whole sample. How do they look? So if you see on the left, the spherical particle has a cubic pore. So each tiny dot that you see here is actually cubic shape, and the pore size is between 3 to 5 nanometer. The right-hand side, MCM41, has a cylindrical pore, and that's why you can see the angle. It goes in the angle and goes in the cylindrical side. And the particle size, as you can see, is about 100 nanometer for both of these. And these are one of the two most widely used. Third one is SBF 15. Three most widely used silica in oral drug delivery. But you can do a lot of different things. So we have been able to make fiber-like structures, very tiny little large pore silica particles, about 30 nanometer, that we're using for glioblastoma delivery. We also have popcorn-like particle structure, so you can induce a surface roughness. Now, the surface roughness provides the additional ability of these particles to be taken up by cells. Like, for example, viruses have surface roughness. They have a surface protein that provides the surface roughness, and this virus-like structure is shown here. So this is actually a silica which has sort of like these fiber-like structure outside that allows them to be taken up by cells more efficiently. A lot of people ask me, okay, how safe is this? Particularly when given orally, silica or colloidal silica has been used in nutraceutical industry since decades. So, for example, your Sensodyne has silicon dioxide. Your nutraceutical tablets, for example, this tablet that they sell in Australia has 300 milligram of silica per capsule, and you need to take 3 capsules a day. So it has been used orally, and it has been approved for oral use in tableting industry for decades. There are many clinical trials going on at the moment using silica nanoparticles, so the future seems bright. However, the challenge is to get the right formulation for the right problem. So what I'm going to do is I'm going to give you some examples of our formulations, the silica particles that we have tried to improve the solubility, permeability, delivery efficiency, site-specific delivery of many, many molecules. So first example is this resveratrol. It's a polyphenol compound from red wine or peanuts. It is anti-cancer, anti-inflammatory. Problem is very poor solubility. Permeability is okay. So what we did is we took MCM48 type of silica, and then we encapsulated that silica into these particles with 30% by weight loading efficiency. So we were able to load up to 300 milligram of this drug into 1 gram of silica particles. Now, how did we achieve that? So the loading mechanism, or the loading efficiency or the capacity is so high is because what we do is we also use a solvent approach. So what we do is we dissolve the drug in the choice of solvent. So in this case, methanol, and we impregnate the soluble drug with the silica nanoparticles. Now what happens is that due to capillary force, that solvent goes inside those tiny little nanopores, and then we evaporate the solvent under reduced pressure. Now, when we evaporate the solvent, what happens is the drug precipitates within those tiny nanopores. And when it precipitates and stays in those tiny nanopores, it will not be able to recrystallize. So crystal growth is actually diminished or reduced over the kinetics is slowed down. And because of that, you have a drug in an amorphous state, and that amorphous state drug is giving you, improved solubility, improved dissolution in this case. As you can see here from this X-ray diffraction graph, the resveratrol is very crystalline. When you physically mix resveratrol with silica, you still see those crystalline peaks, but when you encapsulate using our technology, then it doesn't show any crystalline peaks. So what we did then is we checked that what if we look at the anti-inflammatory activity. So these are macrophages that have that are reporter cells for NF-kappa B. So they have a GFP tag on them, and when the inflammation is high, so when you treat them with lipopolysaccharide, the cells light up green because the green fluorescent protein is going to release when there is more inflammation. So, so GFP percentage, about 50% GFP percentage when you treat them with resveratrol solution. So in this case, the solution means that resveratrol was dissolved in 1% DMSO and then diluted. The suspension is just undissolved drug and then silica loaded resveratrol. And as you can see, the silica-loaded resveratrol has much better efficacy in terms of reducing the NF-kappa B compared to even solution or suspension. Another example is varinostat. Varinostat is an HDAC inhibitor used in cancer. It is orally available. The dose of varinostat is 100 milligram tablet 4 times a day. Very poor solubility, poor permeability. It's a BCS class 4 drug. So if you look at these particles here on the left, the beauty of this is that, as you can see, each and individual particle has exactly same structure. They have exactly same pore structure, and the pores are really, really highly ordered. And that is the important thing, that each and every particle behaves the same when you load. So what we did is we loaded varinostat onto the particle, and suddenly we saw a spike in the solubility. That solubility was further increased when we have amino functionalization. That was further increased when we have phosphonate functionalization. And that also, these formulations also increase the permeability of the varinostat as well. Here's another example where we were able to use floating tablet using silica nanoparticles. We were able to make floating tablets. So the idea was that can we release the drug in the stomach? And we used 2 drugs, captopril as a water-soluble molecule and curcumin as a water-insoluble molecule. And the drug was loaded using our solvent technology into the mesoporous silica nanoparticles. Then it was mixed with HPMC. The polymer here is HPMC. We added sodium bicarbonate and we just did a direct compression. Now, the surprising thing was that we were able to compress those tablets without having any issues in flowability or anything, because the silica itself is directly compressible, and that's really great. You can see the tablets are looking very nice. And then we tested the in vitro release using the in vitro model. So as you can see, all 4 formulations are actually floated. We're able to float, up to 20 hours. One of the things to note is that there are 2 release graphs here. The one on the left, F1 to F4, the F1 is able to improve the solubility, dissolution of the curcumin, whereas the other formulation are not. The thing to notice here is that F1 is the only formulation that has got 1.7 nanometer pore size. So here what we're trying to show is that you can tune the pore size, and that tuning of the pore size can have an impact on the dissolution. So not every single pore size is going to be good for a particular drug. So if you load curcumin exactly the same way using 2.1 nanometer pores, you don't see a lot of improvement in the solubility. However, or dissolution. However, when you have 1.7 nanometer pore size, you certainly see improvement in the dissolution. Now, in terms of the captopril here, F5 and F6, so F6 is the formulation that is made with captopril without using mesoporous silica. And it sort of shows you a very rapid dissolution, finished at 100% drug release in 8 hours. whereas the mesoporosilica nanoparticles loaded captopril compressed into floating dosage form shows you a 24-hour drug release. And that is really good because now what we are able to do is we are able to reduce the dosage. So we are able to reduce the frequency of the dose, and that's really great. Here is some example of another method to load into mesoporosilica nanoparticles, and this is not published. This is fresh off the boat. Is that we've used a supercritical liquid carbon dioxide, a supercritical fluid, and so liquid carbon dioxide was used to dissolve the drug, and then mesoporosilica nanoparticles were added to encapsulate the drug, and we were able to achieve about 30% to 35% by weight loading capacity. So every gram of silica, you'll have 300 to 350 milligram of the drug. And the example is an antibiotic that is given intravenously only, meropenem, because it has a problem of the stability in the gastric fluid, and also it's a PGP substrate. So its permeability is good, but then, the efflux is an issue there. So what we did is we first encapsulated using liquid CO2, the meropenem into silica nanoparticles, and then capped with the udroget and looked at the permeability and looked at the antimicrobial activity. So permeability, as you can see on this graph on the right, A and B, the permeability with silica particles and udroget is much higher compared to the drug. But more importantly, the efflux ratio. So the transport from B to A was also prevented using udroget-coated silica particles that had myrrh in it. And that's really great. So the efflux ratio is only 0.64 compared to myrrh's efflux ratio of 2.72. We were also able to show that the clinical strain 23 of Pseudomonas aeruginosa, the drug that is inside these coated particles, is as active as the free drug. So we are not jeopardizing or none of the drugs degraded, basically. We also use exactly same technology and improve the coating ability using microfluidics technology. And this is also unpublished data where we use a liquid CO2 assisted loading of meropenem, and then we encapsulated those silica particles with microparticles. So say one big microparticle of udroget contains many silica nanoparticles. And once the idea is that this idea where you have particles coming out from one big large particles and sort of giving you improved efficacy. So once udroget it reaches the small intestine, it will start to dissolve and it will start to sort of pop this silica nanoparticles out as well. So we use 3 udroget for that. So udroget RSPO, udroget RS100, and udroget S100. And as you can see, the first 2 A and B here are RSPO and RS100, shows you that zero order release profile compared to myrrh. So if you use only meropenem, the drug is going to degrade in acidic environment within 5 minutes. But here, we were able to retain about 50% of the drug, active drug, in the RS100, about 60 to 70%, and here as well about 40%. But you can see the total drug release is very close to the active drug in all of these cases. So that's a great news. If you look at this graph here, the permeability is also improved in case of RSPO. And we compared that. This line here actually is the verapamil. So verapamil is a well-known permeability enhancer, and that's what has been used here as a positive control. But if you can see the efflux ratio, the efflux ratio of the udroget RSPO coated particles is 0.71, which is very low compared to the meropenem on its own. Of course, it is important to note that when you use the verapamil, the efflux ratio is 0.09, but verapamil cannot be given to the patient who are highly they're very infectious or has a UTI or something like that. And also, it's important to note that the TEER value, the transepithelial resistance with the verapamil, was significantly down, and that could also lead to other pathogens entering as well, but that's just was used as a positive control in this assay. Then we used the same technology coated onto the silica particles, a pH-responsive polymer udroget, and used it for delivery of anti-inflammatory molecules to the colon. Here's an example of prednisolone, where we have prednisolone, the red graph here, water-soluble, kind of reasonably soluble molecule. When you coat it in the silica, the solid dissolution remains the same, but when you actually coat with the udroget, it sort of gives you no release for the first 2 hours here, and then sort of slowly releases over time. The similar trend was seen with the budesonide. Budesonide is also one of the widely used corticosteroid for inflammatory bowel disease, and we were able to show that is possible with our formulation as well. Then we thought that we will try this formulation and test it in vivo using a colitis model. So the mice model that we used to start with was a DSS-induced colitis. So mice were given normal water, healthy mice were given normal water, but the colitis group were given 1.5% DSS, so that's a dextran sodium sulfate. And at the day 7, you transfer them back to the drinking water, and the treatment of the budesonide was started at day 7 and finished at day 11, and then day 12, the animal were harvested. So the idea is that first, the drug will be protected from the gastric environment. It will be released in the intestinal environment. And then at the time when there is no mucus, the silica particles will passively accumulate in the inflamed region, providing that local release. And that's what we wanted. So first thing first, looking at the histology of the colon, this is the distal colon histology. Healthy control mice, you can see this very nice crypt architecture, no ulceration, goblet cells, producing mucin. Epithelium is, very nice, but when you look at this DSS, the crypts are gone. There's no crypt. All the goblet cells are gone. You can see, I don't know if you are able to see here, but these red tiny dots are actually the neutrophils, and those neutrophil infiltration is bad, and that drives the inflammation. When you're using budesonide, you see some recovery of those crypt, but you still see some neutrophil infiltration and ulceration, and crypt architecture is not normal. With the silica particles with and without coating, we saw a similar level of improvement in the histology, which is also seen here in the quantitative analysis when we do the quantitative analysis. It was better than the budesonide, but the difference between the silica particles that were coated and that were not coated was not seen. So we thought that, okay, let's look at what happens at the cellular level and those chemokines and cytokines that drives the inflammation in the gut. What happens to those? So when we look at the mRNA expression of interleukin-1 beta, IL-17, and IL-10, we were able to see the difference between the coated and uncoated particles here. As you can see from DSS, the coated particle shows significantly lower level of IL-1 beta, and same with the IL-17 and same with the IL-10. In the distal colon, the trend was there for both IL-1 beta and IL-17, although it was not reduced significantly, but in IL-10 case, compared to DSS and compared to budesonide in the case of udroget-coated particle, we saw a significant decrease in interleukin-10. Now, interleukin-10 is a cytokine that has double-edged sword. So if there is inflammation, it goes up, but higher level of interleukin-10 can lead to anti-inflammatory activity and downstream pathway activation. So the fact that when these particles were given, the IL-10 was down is indirect measurement of reduction in the inflammation. So far what I've told you is that we were able to load and deliver a variety of small molecules into the gastrointestinal tract using this pH and enzyme and polymer responsive silica particles quite neatly. However, think about another issue that we recently discussed in this review article is this ability of silica particles to deliver biologics. So in this review article, we sort of take this approach where we say, what are the different properties of the silica nanoparticles that can be used to not only improve the delivery of the small molecule, but say macromolecules, and how do we do that? So we hypothesize here and present that the shape of the particle and the surface property of the particle is very, very important. And by changing that surface property, you should be able to improve the permeability and stability of the macromolecules using silica nanoparticles. To do the same, we synthesize many types of large pore silica nanoparticles in my lab right now. And these are the three examples. So these are the dendritic type particles, and these are the dual dendritic hollow silica particles. So you say the smaller pores and then there's larger pores here. And these are like golf ball type particles. So what are the different advantages of these particles? So highly accessible surface area, you can co-load peptides, small molecules. These particles, intelligent synthesis, so we use ethyl acetate as a pore expanding agent. And the last one is actually very interesting. One of my PhD students currently working on it. Look at the loading capacity of the IgG. So immunoglobulin G loading capacity is close to 650 microgram per milligram. So that's 65% loading capacity for such a large antibody. I'll give you a couple of examples. So this is how we synthesize them. So what you have is you have a base, and then you use surfactant, and at 60 degree, you add an organic solvent, in this case, chlorobenzene, and add your silica source. So what happens is that those micelles will be swollen using this chlorobenzene, and those swollen micelles then form a curvature. And that curvature will allow this radial pore coming out from the silica and form this, a very interesting structure. So we were able to functionalize these particles with the silanol group, which is OH, phosphonate group, amino groups, and carboxylic groups using succinic anhydride. In all four cases, we had no issues in the particle morphology. They all look beautiful. Only in the amino functionalization case, this blue graph here, we saw an increase in the particle size. Now, by looking at the TEM, you don't see an increase in the particle size, but the particle size increases due to the aggregation in the DLS. And that aggregation is because of the poor surface charge properties of the amino functionalized particles. And this TGA thermogrammetry analysis is to confirm the percentage loading of these particles as well. We loaded exenatide, a peptide, GLP-1 agonist onto these particles, and we were able to achieve about 40% of by weight loading compared to the MCM41, which has only got about 20% loading capacity. We found that the loading was only achieved at pH 5, which is also an isoelectric point of the peptide, and that's really important to understand that when peptides are at the isoelectric point, the surface charge is neutral, and that neutral charge allows the loading of those peptides into those silicon nanoparticles quite easily. We picked up this phosphonate functionalized particles because we wanted to coat chitosan on it. So the left-hand side image is the phosphonate functionalized dendritic silica particles, and we coated them with the chitosan. As you can see, they become denser, darker. Normally, when you coat, you only see the polymeric structure, but here the advantage is because the pores are so absorptive, it sort of absorbs everything, and this darkness comes from this coating of the chitosan. So the coating does not only happen on the surface here, the coating also happens on the inside porous surface, so that's why the pores are also blocked. Here is a final result for this paper. We used, with the help of Professor Bruno Sarmento in the University of Porto, we were able to improve the permeability not only in the CAGO2 model, but also the triple co-culture model, which also showed that the chitosan-loaded particles had a better permeability. So last thing I want to talk about is something that we've started working on with this cytokine delivery. Now, this is interleukin-22 as an interesting cytokine that has receptors in the skin, respiratory, lungs, fibroblast, intestinal epithelium, hepatocytes, and pancreatic cells. Now, increased production. One of my collaborators worked with interleukin-22, and they have shown that interleukin-22 is able to produce very high quality insulin when the beta cell function is a problem, so in the diabetic patients. It also enhanced the protection of the mucus producing cells and elevate the mucus production, so it has implications in the IBD. So what we wanted to do is we wanted to see if we can improve some of the properties of the interleukin-22. The challenges of the interleukin-22 are very similar to any protein or peptide, any protein. The stability is a problem, permeability is a problem, off-target effect is a problem. And all of those challenges we went to see if we can use silica particles to overcome those challenges. So the first challenge was the permeability. So we were able to load the interleukin-22 with 100% loading efficiency into silica particles quite easily. We were able to permeate some of those from A to B. However, if you look at the apical chamber, the IL-22 loaded nanoparticle, which is your silica particles, actually stabilized the interleukin-22 for up to 24 hours. Whereas the only IL-22 was percentage was decreased, so it started to degrade. So we are investigating what's happening there, but that's certainly something interesting that we found. Then we used LS147 T cells, luciferase cells, to look at the STAT3 phosphorylation induced by IL-22. For the first 24 hours, the results between IL-22 and IL-22 nanoparticle look very similar. But after that, the interleukin-22 loaded nanoparticle performed better, because after 24 hours, the release of the IL-22 from the nanoparticle was still probably happening, and the cell was still producing the STAT3. So that was really good. Then we used, again, DSS model to test whether locally delivered IL-22 can induce the anti-inflammatory activity, and whether the nanoparticle loaded IL-22 will have any impact on that. So DSS treatment for 5 days, and from day 4 to day 10, we gave rectal delivery of IL-22. And then that rectally delivered IL-22 was also compared with the positive control where we had IP IL-22 given without nanoparticles. So one other interesting thing that we observed is that compared to the blank, the IL-22 rectally given nanoparticles decreased the colon weight to length ratio. Now, that is very important in assessing the inflammation. And then what we wanted to see is, okay, what happens at the molecular level? So the molecular level, interleukin-10 was up by IL-22. That shows that the inflammation was there, and then as an anti-inflammatory cytokine, the IL-10 production was increased. And that was also accompanied by the interleukin-22 binding protein increase, which told us that the IL-22 that was delivered via nanoparticle was actually bound to the receptor. This is not a PK, actually. This is just a one-point analysis of the blood where we looked at the any interleukin-22 in the blood after 24 hours, because our ethics protocol did not allow us to take lots of time points, because the colitis mice are very weak, and that can lead to ethical issues. But at the 24-hour time point, the IP injected nanoparticles, we saw some IL-22, but we saw nothing in locally delivered. So finally, the idea is now what we're working on is that delivering these and other proteins and inflammatory proteins orally using this particle, but then using the layer by layer coating technology to coat them and then deliver them. I just want to thank finally my team who did all of these work and the funding bodies, NHMRC, Mater, and I just wanted to remind everyone that as part of my other role as the president of the CRS, we have a CRS annual conference on 23rd November. Professor Justin Haynes, who is the professor in Johns Hopkins University, is going to be a plenary speaker. So if you want to register, the abstract submission unfortunately is closed, but the registration is still free until the last day. So please go to the website crsaustralia.org and register if you want to listen to those talks. Thank you so much. I'm happy to answer any questions you guys may have.
Yeah, thank you, Professor Ahmed. Now, if there are any questions, if anybody want to ask anything.
Professor Popat, this is Anirudh here. Excellent talk. I just have one kind of doubt, not exactly doubt, one query. Have you studied the pathway of absorption of these silica nanoparticles?
We have. So so there are a couple of things. So silica nanoparticles are absorbed through a couple of routes. Okay, and one of the ways they're absorbed is opening the tight junction. So silica nanoparticles are kind of permeation enhancer. So it sort of opens the tight junction transiently for depending upon the dose, and then it sort of goes through there. If the silica particles are not, if the dose is not enough to have that effect, silica actually degrades into the silicic acid, which is just going to come out in the urine. So the pathway of the degradation is quite simple there. The other pathway is that it is due to the size, they are also taken up by M cells. So they go through that system where they're taken up by M cells and then go to the bloodstream and then degrade to silicic acid. Yeah.
Thank you. Thank you.
So thank you, Professor Ahmed, for Yes, Arihant. You have a question?
Yes, sir. Yes, sir. So, sir, thank you so much for your presentation. My friend was also working under your lab, Bhalchandra More, last year. He was already told many things about the silica nanoparticles. So, I just wanted to know the surfactant. What should be the surfactant property you are choosing? According to that, they can form as those self-assembling with the silica.
So, see, surfactant, you have a lot of choice. Typically, you go for your surfactants such as CTAB or CTAG, but you can also use surfactants such as F127. So Pluronics have been very successful in forming the large pore SBA15 type of silica particles. And as we all know, Pluronics are FDA approved up to a certain dose. So Pluronics is another surfactant that you can use. So you have endless choices over there. What you're looking for is that the surfactant that can form self-assembly relatively quickly. So you don't want a very high concentration of surfactant, but it forms that self-assembly, say in 20 minutes, 30 minutes, as soon as you dissolve them. So I think that's what you're looking for.
Thank you, sir.
So thank you, Professor Ahmed, for giving a talk on the silica nanoparticles and how they could be used for oral delivery. And thank you for sparing your time for the talk. Yeah.
No worries. Thank you, Deepak. Thank you for invitation.
Yes. So our next speaker is Professor Harish Kumar Madhyastha. So I take this opportunity to introduce Professor Madhyastha. He is a faculty at the Department of Applied Physiology, University of Miyazaki, Japan. He is holding 2 PhD degrees. He started his career as a scientist in Murugopa Chettiar Research Center, Chennai, where he was a team member of Spirulina Technology Research. Later on, he pursued postdoctoral research at Miyazaki University that culminated in a faculty position at the Department of Applied Physiology at the University of Miyazaki from 2006. His current research interests include generation of novel platforms for delivery of nano-sized biomaterial payloads for regenerative diseases. His academic credentials are credited with H value of 27, Clarivate analytical cumulative impact factor of 167.5, and RG score of 31.8 with 74 Sci Index publications and 6 international patents. He is also actively involved in many international projects, including ongoing Indo-Japan scientific and academic collaborations. He is a fellow of Biotechnology Research Society of India, fellow of Royal Biological Society London. He is an officially recognized Indo-Japan academic spokesperson of University of Miyazaki and engaged in outreach programs to further strengthen the cohesive relationship between Indian academicians and University of Miyazaki, Japan. So with this, I invite Professor Madhyastha for delivering his talk.
So thank you, Professor Deepak and Professor Jadhav for the kind invitation of this project, this seminar and virtual conference. Special thanks to Professor. Sun rahe hai na?
Yes.
Yeah.
Sorry.
Okay. Yes. Special thank to Professor Deepak. Professor Deepak, can I have the first slide, please?
Yeah, I share from here, sir. Just give me a second.
Yeah. Fine. Yeah. Today I'm going to talk with the surface modified colloidal nanoceuticals. It is gold quercetin conjugate during the mimic wound healing. Why mimic wound healing? It is a in vitro models to study the whatever the drugs which we are interested for the regenerative medicine in the future course of action after particular in vivo studies. My name is Madhyastha Harish Kumar. This is my email. So please take down email and have the continuous interaction after this conference also. I am available 24/7. Next slide, please. So I'm going to talk with this background of this my paper. It is long back we published in the Colloids and Surfaces CA in 2018, where we have synthesized the quercetin gold nanoparticles and studied its basic actions. All properties, preliminary biological characterizations already published in this work. Next. Yeah. So in this one, what is the functionality nanoparticles? It is this, we have the core shell, either it is of the metal or silica or liposome or whatever it is. Then we have the surface modification with the various molecules or a propertized materials or some antigens or some the shape recognition modules or some the signaling for the biotheranostics agents or biocompatible drugs, linker molecules, linker polypeptides, linker proteins, so many things we can have after the proper tuning in the surface. Next. So we hypothesized that when you have the core AU with the surface modified to quercetins, if you treat the fibroblast cells, whether it is have the activated fibroblast or not. Now, what is the particular biointerface mechanisms? That black box we wanted to know because, next. So my area is mainly focused on the regenerative medicine, particularly the diabetic and normal wound healing systems. We have the diseased skin here, whether we can form into the normal skin after applying with the surface modified nanoparticles, which are particularly conjugated with many useful drugs, either of the natural drugs or the synthetic drugs. So we hypothesized that AU quercetin nanobiomolecular interface during the in vitro wound healing has some effect or not. Next. So in the present study, we have depicted the regulatory pathways involving the wound healing process. And here we confirm that influence of the microRNA 21, SMAD2, 3, SMAD4, SMAD7, and also the wound healing process treatment of this particular conjugate as a therapeutic agent. This is in vitro studies. In vivo studies are going on. What is the quercetin? Quercetin belongs to flavonoid family. It is known very well. It has a 3 rings and 5 rings hydroxyl groups. And the phytochemical components in it have the remedial effects on the wound healing, particularly. Of course, it has several other properties also, but my interest is whether it has a particular effect on the wound healing or not. Basic problem with the quercetin or any kind of flavonoid is it is bioavailability is very less, and but biocompatibility is more, and it has the high elite property of the cytotoxic effect, and it is not. And efficient drug delivery vehicle can be proglaminated with this quercetin. Of course, AU is an antibacterial agent because it is a metal. Next. Fine. What is the wound? It is in that medical term, it is a pathological state where the tissue becomes separated or destroyed. That is one thing, and resulting the loss of the body or substance associated with the function impairment. The reverse function of this one is healing. Of course, we have to gradually modify those defect surface into the healing with the normal shape. Otherwise, it doesn't have proper functions in the body functions, or a tissue damage will be there. This is a schematic, a wound, and there is a wound, and bring back into the original slice of original tissue as it is in the original shape. Next. So there are several classification of wound. One is the physiological wound. It is called the menstrual cycles and lochia. The lochia is the postpartum bleeding. When there is a childbirth process is taking place, there is a postpartum bleeding will be there in the body. It is called a lochia. It has to heal within 24 hours. Otherwise, there is a severe postpartum bleeding system, and ultimately patients may die. Another one is the pathological. It is ulcer, brachial plexus injury, diabetic neuropathy, brain aneurysms, and ischemia. This is the pathological internal wounds. And there is a pathological external wound like skin injury, bone injury, burn injury. The burn injury has both two types. One is the closed wound and open wound. I am particularly interested in the open wound, which is exposed to the environment. It is a dermal wound systems, particularly with the diabetic conditions and non-diabetic condition. Next. So this is the types of wound. It is the internal wound and external wound, as said in the previous slides. It is what is the implications of this wound? The internal wound has the impaired circulation leading to the venous and arterial ulcer, ultimately the gangrene and maybe amputation in the later stage. And the neuropathy triggers the foot ulcers and Charcot joint disease. Of course, we lose the sensation of the tissue ends like fingers and other things. Another one is called tissue inflammations, the genesis of the tissue degenerations. Of course, these are all the internal wounds, pathological wound. The external wound is the closed ended. There's a lot of bleeding and large bruises, chances of the bone fractures, and compartment syndrome in the body, particularly the diabetic patients. And the open wound has the good platform for the infections, loss of functions, scar formation, and decrease the body deformity. If it is a permanent, unhealed or improperly healed, this one, and of course it has the bad appearance. None of them want a bad appearance in the outer body. The next. So answer is the coronated metal. Can we have some coronated metals or not? Of course, this is the metals system. We have the coronated systems with the natural systems or from the drugs or whatever it is, which is of the particular interest. Fine. So next one is we predicted that the quercetin, as I said earlier, the quercetins has the alanine 72 and alanine 142, glycine 142, and alanine 142. Which has the particular binding site with the gold metals. This is the bioinformatic predictions, and we did the functionalization studies by the time, where you can see there is a clear nano-sized increase in the TEMs after the functionalization of the quercetin with the AU metal. Fine. Next. So fibroblast cells are the major one of the target cells which has the cumulative and collaborative functions with various cells, particularly with the keratinocytes, platelets, microfibroblast, capillaries, and macrophages. through the various signaling pathways, TNF alpha, growth factor mediated, angiotensin, and TGF. Now, I am interested in the TGF beta and how the fibroblasts communicate to the keratinocytes and keratinocytes communicate back to the fibroblast to the IL-1. Next. So this is the after the treating with the self-operative, the untreated wild cells have the particular cells. Of course, it is a very small one. And AU treated cells, the cell has AUNP has a little toxicity towards the fibroblast morphological cells, but AUNP, when coronated with the quercetin, it has found back into the normal cells. Next one. And endpoint toxicity shows that the various doses of the AUNP. Here we considered AU microgram per liter in AU ppm level as a 0.05, 1, 5, and 10. Of course, 10 has the severe toxicity level to the cells in the terms of the viability of the cells. It is decreased, but 5 ppm has increased. The AU quercetin conjugated cells gradually increased, but AUNP alone shows the decrease in the cell lines. This is the time period. So here we confirm that 5 ppm of the cells at 8 hours time is not toxic to the fibroblast cells. Next. So, as I said earlier, we wanted to know the communication between the keratinocytes and fibroblast cells. With the here, fibroblast with the TGF beta and ET1 converts into the protofibroblast with the elastins, and with the final molecules of the TGF beta, it converts into the myofibroblastins, which increases the tension of the newly formed tissues. There's a tension rate which is more than 3 index is the good reformed tissue. You can see in this molecular cellular medicine of the 2017, I explained what are all the how tensions with the treatment of these various drugs has increased in the newly formed tissues through the communication from the keratinocytes and fibroblast altogether. Then these are all the pathways. And next one, yeah. Here we did that TGF beta, both protein as well as the Western blot, the protein as well as the gene cellular gene level with the different concentration of the AU conjugated quercetins. Of course, here 10 microgram per liter, 10 ppm of the AU shows the little increase, significance increase than the other cells. It is the with the gene at RT-PCR studies, and but the protein did not show the much increase. But of course, in the 10 ppm level, it has significantly decreased, but not severe like the quercetin alone. Next. Is that video, the co-culture cell culture, is it working? If it is that, please play that one. Yeah. Yeah. Here, what we have done is that this is the wild fibroblast where the TGF beta plus. It is the normal fibroblast cells. Here, we have two kinds of the cells. One is the normal fibroblast, which is gray in color. Another one is TGF beta minus. We knocked down with the gene editing tool and labeled with the green fluorescent proteins. And we co-cultured the both cells together and did the migration pattern in live conditions. Here we can see that this normal fibroblast in the plus condition moved fast, whereas in the green color one, which is the minus TGF minus condition, did not move fast, which confirms that TGF beta is necessary for the cell migrations. Why? Because of the various binding and skeletal proteins like vimentin and actin molecules. This is the TGF beta minus where the actin and vimentin is completely denatured. This is the control cells. Fine. This we have seen. This we published the materials today in 2019. Fine. Next. And this is the. So once we confirm the TGF beta is necessary, and does it have any SMAD crosstalk, which is that the SMAD is another transcriptional factor molecule proteins which has important for the wound healing. And how it is then is the TGF beta, there are three families, beta 1, beta 2, beta 3 has the receptor binding protein with the receptor in the membranes, which increases the MAP3 kinases up to 7, and SMAD 2 or 3 or 1 or 5 or 4 or 7, 7 is the inhibitor of the SMAD 7, increases the migration of the SMAD 4 into the nucleus and transcriptional regulation taking place in the nucleus by the induction of various genes. Next. And we did the same with the what is the SMAD2, SMAD3 and SMAD4 protein analysis. In all cases, 5 ppm of the gold quercetin treated cell shows the increase of the SMAD3, SMAD4 and SMAD3, but not SMAD2. But SMAD2 is in still lower concentration. 1 ppm has the highest activity. Sure. Next one. And if it is increased in the SMAD2 and 3 and 4, if it is increased in the nuclear total protein after the treatment of the AU gold quercetins, where it has to go? Either it has to degrade in the ubiquitous proteasomal degradation levels, or it has to migrate into the nucleus to increase the various gene transcriptional levels. Yes, we found that this is a SMAD2, there is no changes. SMAD3 has gone into the nucleus. Here you can see, and light pale green colors, and SMAD4 no. So SMAD3 increased, SMAD3 migrated into the nucleus. But why SMAD2 and SMAD4 did not go into the nucleus? Because of the overexpression of the SMAD7 in the 1 ppm level of the gold quercetin nanoconjugate treated cells. Because the SMAD7 is the inhibitory actions for the, or it is called a speed breaker for the migration or the transmigration of the SMAD2, 3 and 4. So this is the reason that while increase of the SMAD7 did not have any effect on the SMAD3, but it has the cumulative effect on the inhibitory action on the SMAD2 and SMAD4. Next. So complication in the wound healing, as I said earlier, TGF beta and IL-1 is most important. It has the transcommunication between the keratinocytes and fibroblast cells. We hypothesized that. Next. Hypothesize that if diabetic condition is there, it is a proven data that TGF beta increases in the diabetic conditions. So that if there is an increase in the diabetic condition, TGF beta oversecretion will be there. It has the ultimate effect on the keratinocytes, and keratinocyte growth factor released into the cytoplasm through the IL-1 increase. And if there is an increase in the TGF beta, it is epithelialization of the new cells, new tissue will not be taking place, so that healing will not be proper. This is the hypothesis, complications, complication with hypothesis. Next one. So we put ourselves the question that does it have any applications on the cell migration pattern? Because cell migration is the initial or the first stage of the cell tissue regeneration in the wound healing. Fine. Next. So once again, we put the miRNA level both at the miRNA 21 and because in earlier publications we proved that miRNA 21 is overexpressed in the diabetic in the patient's care. In the patient's cohort studies, we proved that one and published long back. And we wanted to know whether it has any implication on the miRNA 21, and this is the result where it has, yes, 7 ppm, 10 ppm has the highest activity of the miRNA 21 total expressions. Next. So this is the mimicking wound healing stretches. This is the control, 0 hours controlled and 8 hours controlled. 0 hours controlled and 0 hours controlled with the both control as well as the AU quercetin treated cells. There is no migration of the cells, whereas in the after the 8 hours conditions, AU quercetin nanoparticle treated, there is a clear migration of the cells. Yes. Next. So this is the Z-Stack videography instrument. What we are taking the live video cells. Next one. In the first one is this block is the control one. This is the quercetin alone treated one. This is the quercetin conjugated treated one. So if you see that migration of the cells in all three cases, and here cells which are treated with the AU quercetin group shows the highest migration of the cells, because here wound is almost completed. Here there is still gap is there. Quercetin treated, of course, quercetin alone doesn't, it is almost equivalent or little less than the control cells, whereas in the quercetin gold conjugated cells has the edge cell migration. Why? We asked ourselves question, why it is so? Because these edge cells in the wound edge cells has the highest stressed cells because of the highest reactive oxygen species production in the cell. In the edge cells, because it is directly exposed into the oxygen tension in the environment or in the media. So this quercetin is the antioxidant. We hypothesize that quercetin being an antioxidant is a preventive action for the further degeneration of the cells, edge cells, which is directly exposed into the environment or the oxygen tensions. So that quercetin in one way helps as the antioxidant molecules, and which is being captured by the AU metal. AU metal is a good antibacterial or anti-stress molecule also, so that this helps is the lowering the inflammatory molecules. So we proved that AU quercetin conjugated system is better than the quercetin alone and control, and as well as in the control. Next. So in nutshells, that in the fibroblast cells, the conjugations in the fibroblast cells increase the SMAD complexes, and which prevents the 2, 3, 4, which prevents the migration to this one, which increases the mRNA-21, and would independently induce the cell migration, because there is no except pre-mRNA-21, none of the other speciation of the microRNA-21 did not increases. Of course, it is already published just some 3 weeks before we published this work. So next one, take-home message is, I always emphasize this one. Of course, we do in the laboratory, we do so many biomechanical or biochemical and pathway signal transduction mechanism, then all other things are good for the academic one. But if it is come to the society, if it is come to the patients, always it lingering into my mind that so many nanomaterial based products are available in market, and it is been authenticated by FDA also. So my question is, if it is accumulated regionally in the tissues or in unknown portions, what will happen? Of course, there are many publications or many hypotheses are available. It is excreted either in the urine or in the sweat or in the some other sweat and urine is the major things. But if not, if it is after all, it is more than some 10, less than 100 nanometers size. If it is going on accumulating in some specific tissues, what will happen? Our body is, tomorrow our body will be a gold mine or a silver mine or a zinc or whatever it is, metal mine. Once all our scientists, we should think about that proper excretion and should not accumulate in the body. That is the one, my ultimate aim of all these nanomaterial studies. So if you want to read further, this is the latest publications we published in RSC Advan just 3 weeks before. So thank you. Arigatou gozaimasu. This is Japanese word. Thank you.
So thank you, Professor Harish, for giving the talk. So now any questions?
Sir, one question.
Yeah.
I'm Anirudh from Pharmacy Department only. This is an
Yeah.
excellent presentation.
Thank you.
Do you have any comment, because you are a expert in this field? Do you have any comment on the availability of the tumor matrix which is in the wound area, and the effect of the non-availability of tumor matrix, sorry, not tumor matrix, tissue matrix, for the migration and the wound healing process? That, as that means, to be more clear, that the cells are there, that this fibroblast, keratinocytes, their precursor cells, everything is there. We are giving treatment with something. And what is the requirement of a extracellular matrix in that wound bed for proper wound development, means wound healing?
Yeah, because ECM matrix, especially the MMPs, so that is most important for the proper anchoring of this fibroblast and keratinocytes particularly. These two are the anchoring cells. That is most important. MMPs as an anchoring molecule is most important for the proper migration of the cells. That is most important. So in one of our previous publications, we have done what is the relationship of the particular another on the fluorescent protein. It is called phycocyanin. We did the MMP's expression in the artificially created wound bed. The 2D cultures, we cultured the keratinocytes, fibroblast, and macrophage together. Then we applied that one and we did the MMP expressions. And MMP expressions were very much increased when you add particularly this some kind of natural product based medicines.
Okay. Thank you.
Sir, this is Swati from Pharmacy Department. Sir, just a small query that you recently told that you have a co-cultured macrophages, fibroblasts together. Can you suggest something that if we have some polymeric bed or so, and if we want a co-culture system, then how we can proceed with that? Apart from transwell, other than that, if we can have any option.
No, you can have this collagen coating to the if you are doing with the 2D cell cultures, we can have manually, we can coat with the collagens, one thing, and gelatins, second thing. Nowadays, one German company, it is called Mu-Ibid culture vessels are available. In that one, if you have the tailored made coating also we can have, or you can have layered coatings, or you can have the compartmentalized coating. I want left to right, 1 centimeter of the gelatin and 1 centimeter of the collagen. Then you can, or you can have 50% volume by volume or weight by weight based. This one also we can have.
Okay, sir. So one more doubt I have.
Yeah.
Sir, as TGF beta is important for migration and myofibroblast differentiation. Sir, at times myofibroblast differentiation may lead to scar formation
Yes.
also. So
yes, I expected this question.
Sir, is there any specific place like where we can say that, okay, now after myofibroblast differentiation, this will stop? Is there any specific marker or so? To be simple, how we can confirm that if we are targeting scarless wound healing, then what should we check or so?
So scarless wound and scar formation is the one thing is myofibroblast overactivation is the one phenomena. Another one is, and this is the last, scar formation is the last stage of wound healing, right? In that one, major players are the keratinocytes. Keratinocytes are there. Because of the keratinocyte growth factors, overexpression of the KGF, keratinocyte growth factors, 1 particularly, which has the dynamic role in the scar formations. We should have a kind of what you call time-lapse drug delivery systems. Time-lapse drug delivery systems. Suppose if you conjugate, of course, quercetin helps in the fibroblast migrations. But quercetin may not be a good agent. May not be, I don't know. It may not be a good agent for the KGF productions. So that in the tertiary or the bilayer or the tertiary layer of your conjugations, you have some molecules which inhibit the overexpression of the KGF or overactivation of the keratinocytes. In that way, we can control the scar formations.
Okay, sir. Thank you, sir.
Yeah.
So thank you, Professor Harish, for sharing your insights and sparing your time.
Thank you. Thank you.
So with this, we close our morning session. And in the afternoon, we will start at 2:30 and we have 2 talks scheduled. One is by Professor Sharma from Naipur Mohali and another one is from Professor Padmavi Devrajan from ICT Mumbai. So let's meet again in the afternoon session. So please join at 2:15 sharp. Thank you all for joining in.
Thank you all. Thank you all. Thank you.